Hey, it's Peter here with my FSHD, and like I tell you about before, you know it's a big day when we got the CRISPR goddess with us.
A big day. Just means this is going to be a long session, huh?
Well, we've taken care of that. We've had our
90 slides, but don't worry, we're going to go fast.
90 slides.
Something like
that. Is
that.
there that much to talk about?
I guess. We parse things out.
Well, you know, we try to be complete.
We try to be thorough.
Right.
Yeah.
Try to cover all, but the thing to remember is we are the harshest critics on ourselves, I would say.
Yep.
And by actually publicly putting this out there and putting our names behind it, which no one else has the stones to do. Trust me, a lot of people have these similar opinions. They just don't want you to know it. And they'll pretend that they don't when they meet these companies in person.
When we go to trial, we expect somebody out there to do the same for us.
Exactly. Again, I have looked it up. There's, as long as you tell the truth, you're good.
Yeah.
Right? So if we're ever,
or dead. But you're good.
We're all about the truth. And we're scientists.
Yeah.
I like to just think we are condemned to our fate because we're just scientists at heart. And we ask questions. And we are...
innocent questions.
Well, no, it's true. Well, when something doesn't smell right, we're like, what the hell is that?
Can't let it go, so.
Yeah, right? You pull out the stove and you find the turtle that you've been missing for 6 weeks. Right? You don't just leave it there and open.
Who hasn't done that? Exactly. Come on.
Well, washing machine.
Yeah.
Anyway, the point is, we like to get to the bottom of problems. We like to make things better. And, and we also want to explain to all of you all out there, just so you understand maybe some things that are not being explained to you by foundations or companies that may not want you to know certain things.
Yep.
Or maybe they do and they're just too dumb to tell you. I mean, that's the golden, maybe that's the silver lining. They're just a bunch of idiots.
It's always possible.
Okay, so we're going to leave that as an open possibility.
Yeah.
And we're definitely... Well, I know I'm the dumbest person in the room right now.
He always says that.
Yeah, but I mean, I'm sitting here with you. It's just... But anyway, I think we got an interesting podcast because we're going to be talking about questions that have come up about the recent International Research Congress for FSHD, whatever they call it now, that was in Chicago that we did not go to.
Yep.
But just about every question I had one question about souffle, and every other question was about Epicrispr. Because evidently we can pack up the lab and go home and go fishing because FSHD is cured.
All right.
Cautiously optimistic, but we I really need the clip of Lionel Hutz. You know Lionel Hutz?
Oh, yeah.
Yeah, the Simpsons lawyer who says, I'm not allowed to tell you that I can guarantee you that you're going to get a lot of money, but I guarantee you you're going to get a lot of money. So cautious optimism that seemed to be interpreted on Facebook and social media and everywhere as the job is done, hallelujah, release the doves. And you know what? I hope it's true. But you know what?
It would take a miracle. Let's see.
One miracle at a time, as Neil Camarda always says, one miracle at a time.
Does he always say that? I like that.
Yeah. Actually, I had a meeting with him today. And he breaks that. He's got time is muscle, one miracle at a time. And
no more than 3 impossible things before breakfast. Or maybe it's 6. I don't remember. That's Alice in Wonderland, though.
Well, Neil works with Chip, and I'm sure it's an interesting rabbit hole. All right, let's go to the presentation. All right, so here it is. It's the IRC on FSHD with their co-opted light bulb. I like Kate's better.
But we won't get into that.
We won't get into that. I digress. We're going to be long enough. Okay, 33rd, Dan Perez started these. It used to be just a single day. All the science was done in the morning, and then he had his international lab meeting in the afternoon where nobody really wanted to talk to each other because everyone would steal each other's ideas. But it was such a small field, and it grew to now it's multiple days, tons of science. And honestly, though, what I heard from people that attended, it's basically just become an industry show. Very little research. It's really, I don't know, people are very unimpressed with the research, but it's an industry show. Yeah. It's like a trade show now. So whatever. But we're going to be talking about it. But that's good, right? Because it means a lot
to talk about.
A lot of companies are in the space, which is an important goal.
Yep.
but a lot of some of the top researchers were not there for good reasons. So let's go on. So what came out of this? Well, I'll tell you, first of all, the first question I got was we've been telling you about souffle, right? So souffle, I got to remember to look at the right spot now. So souffle is, they just got in the game. Actually, they've been in the game for a couple of years on siRNA. They're really well backed. They got like $3 billion in backing, and that's with a B, and coming out of MIT. Some really fantastic stuff. And their idea with their siRNA is they have a novel peptide that targets the siRNA really well to skeletal muscle. So they can use a lower dose, much more efficient, and the durability of the effect is going to be longer, like 10 to 12 weeks. So and they're pretty confident that they're going to be going forward with a subcutaneous injection. They're going to do IV right now, but their sub-Q is coming close enough that they actually discussed having a sub-Q clinical trial. So a lot of things. So that caught some people's eyes. Actually, my man Baja Blast picked that up, an admirer of yours. The man's got good taste. And so he asked that question. So he said, so there's not much more to talk. I got to be careful what I say about them, other than, what he was kind of curious is it could be like an EpiPen or something. I don't actually know how sub-Q is going to be delivered, but you can imagine like a GLP-1 pen where you hit yourself every 10 weeks. That would be fantastic.
Yeah.
But you know what? Every other company out there is trying to get a sub-Q as well. They just haven't, I'm willing to bet. Okay. But what we really want to talk about is the big news, breaking news. Epicrispr reports first clinical evidence of very carefully worded press releases, by the way. Evidence of increased lean muscle volume in patients with FSHD following treatment with Epi321. That's from the FSHD Society, who is a partner with Epicrispr. And we understand what that partnership means. FSHD Global Research Foundation reports first clinical evidence of increased lean muscle volume in patients with FSHD following treatment with Epi321. More breaking news. Okay. So this is really fantastic. My email was lighting up and thought, hey, actually, we wish everybody success. Fantastic. I can't wait to see the data.
I can't wait.
I know it's gonna. I'm prepared to be blown away. Yes, I'm prepared to be blown away because that's pretty impactful. I mean, it's all I went on Facebook and that's all I see. Everybody, oh, thank God, this is what I've been waiting for. Whatever, amazing. I hope we can afford it. I mean, when's it gonna be available? Everybody's lining up for this, and that's and that's what you want, right? Assuming it works. And they couldn't possibly have this kind of press if it doesn't work.
It's never happened before.
It would be the first time ever.
First time in the history of the world.
And especially FSHD, better than expected.
Oh yeah, we haven't heard that before. Okay, let's go.
By a partner of a company.
Yeah.
My goodness. But you know what? I also get a lot of questions about AI. And so, I was thinking about it. Since everybody's just AIing things, and they're just going to do AI as to, they don't need to listen to the podcast anymore, which is why we need our witty banter.
Yeah,
because
AI
doesn't do that. So I just asked AI, what is the job of a scientist? And this is what AI told me. Okay.
Let's see how many we actually do.
The primary job of a scientist is to study the natural and physical world to discover new knowledge, solve complex problems, and explain how things work. They use a systematic, evidence-based approach called the scientific method to turn curiosity into proven facts.
Proven facts, huh?
Well, that's what scientists are supposed to do.
What about models?
Well, you make models that turn, but at the end of the day, you say it's a proven
facts,
based on the end, you're trying to get to.
See, already you can tell AI isn't 100%.
Oh, they're going to crucify you. You think it's bad enough that we're doing a podcast on science and criticizing our colleagues?
I guess.
Although I would point out, we don't want to say too many bad things about AI. Trust me, on this podcast, right? Because AI is, A and I, right?
Those data centers will chew us up and spit us out.
Anyway, what are the core daily responsibilities? Observing and questioning, okay? Noting patterns in nature, technology, or human health, and asking specific measurable questions about why or how they happen. So observing and questioning.
Suppose they mean questions with measurable answers.
Well, what, you're going to limit your questions? I mean, you figure out measurement, you define to it, but you're still going to ask the question, right?
Measurable questions.
Forming hypotheses, developing educated guesses or theoretical models to explain those observations. Conducting experiments, designing and executing rigorous tests in laboratories using simulations or conducting fieldwork to test hypotheses. analyzing data, collecting information, measurements, and evidence, then interpreting the results using advanced mathematical and statistical models. More models. Shared findings, writing detailed reports for peer-reviewed academic journals, and presenting discoveries at global conferences to advance the broader scientific community's understanding. And to secure funding, writing grant proposals to pitch ideas to governments or private organizations to get their money needed to run the research.
That should go first.
So that's me first. Because none of this other stuff happens if you don't have any
Unless
money.
you're us.
Unless you're us, who somehow are able
Somehow.
to get all this stuff done with no funding.
Collect coins
Well,
in grocery
I'll tell you,
stores.
I remember ages ago, I told Chip and Neil, I said, I'll put our unfunded lab up against your $100 million. I bet we win. And you know what? I'm still betting. I'm still taking that bet. But this is what I'd say we're doing the job of the scientist. We are observing and questioning, and that's all this podcast is. We're observing and questioning and forming hypotheses. We're looking at people's experiments, interpreting the data, analyzing the data, and you know what? And we appreciate everybody that shares their findings. That's what we're doing. So what's the job of a patient foundation? To improve health outcomes and support those affected by a specific disease. Disease foundations typically focus on the following. Okay? Now, I'm hoping that some of these foundations do an AI search and say, what is my role?
Whoops.
Because some of them, or maybe AI is wrong.
Partnering with companies, making money,
fund research to
padding
advance
their own pockets.
This is, foundations are started this way.
Yeah.
Fund research to advance understanding and treatment of the disease, provide education and resources for patients and families,
All right.
advocate for policies that support disease awareness and funding, facilitate support networks and community engagement for affected individuals, raise funds through events and campaigns to sustain their initiatives,
Definitely.
collaborate with healthcare professionals and organs to enhance care.
All right.
Boy, that I would actually agree with AI.
Yeah.
I think A is very I. This whatever. I think whatever. I don't know who this is. This Claude or Siri? I don't know.
I don't know.
I'm checking that. Let it Google. Anyway, good answer. All right. What does a biotech company do? Now they're getting into trouble. To advance health and sustainability, biotechnology companies focus on the following. They develop innovative medical therapies and vaccines to treat diseases. That is true. They hope to, right? They create genetically modified organisms for improved agriculture. Well, that is true. Not in our space. In our space, no one will spend a dime on creating tools. Conduct research to understand biological processes.
Agricultural yields.
I know. And disease mechanisms. Actually, they don't give a crap about disease mechanisms either, as we will see. I would say understanding processes they leave to the academia, and if academia does a good enough job on all of these, then biotech is interested.
Yeah.
But as you well know, from your experience
Yes.
as a consultant,
Yes.
when you start asking questions about mechanisms,
No, they don't want to hear
they
it.
fire you.
That's right. That is the slow way to nowhere.
How does this work? That
Who cares?
You know what? We could test if this is actually working.
No.
Grace, we no longer want you to attend our meetings.
Don't
Heresy.
call us. We'll call you.
Heresy. Can you believe? I know. Chuck, you can see it. It's like a far sight. Little. I was like, can you believe that she actually wants to know how this works? Oh my God! What a rube.
Not a rube.
Produce biofuel. Well, we'll skip that part. Implement biomanufacturing techniques for efficient production of biological products. Absolutely. In fact, that's this is actually one of the biggest things. If you get in a drug that works, but you can't make it to scale good enough or pure enough or safe enough, it doesn't matter. So that's actually a huge thing. Collaborate with academic institutions and partners for research development. Well, basically, they siphon off our knowledge and our resources. Nothing says making money, so I think AI has missed the point because they don't seem to think that actually biotech's job is to make money. Actually, they're businesses, and their number one job is to
Which supersedes these other interests.
Keep their investors happy by returning on the investment, which may or may not mean creating a drug that works.
Right.
It means telling them that you've created a drug that works. and showing the illusion that your drug works,
Yeah.
and then selling your company before they find out it doesn't work.
See, before you get involved in this, as we have been, you don't realize that. You think that this is how they make money. Actually, no.
Right.
Not at all. In fact, this
It's
takes
AS.
definitely plays second fiddle to the primary goal. But anyway.
Well, see, again, we didn't realize how altruistic AI is. I thought AI was going to tell you how it is.
Idealistic.
Yes,
This is the most idealistic stuff.
mean,
Yeah.
it's
Wow, so warm and
artificial
fuzzy, my
idealism.
God.
Maybe that's what the I is. You know what? It says AI. It's artificial idealism.
Yeah, there we go.
Because it sure as hell isn't how biotech
I always thought
works,
they
or
were cold,
pharma.
these
Yeah, I thought the computer, but
these algorithms.
yeah,
No.
well, you are as
Warm
good as
and
the
fuzzy.
information going in. So anyway, all right. Well, I thought, okay, that's interesting. So so we're not intelligent. We're not artificial intelligence. We're not real. But so what do we think? So we think the role of a scientist is to find the truth or the correct answer to impactful questions, develop hypotheses, create tools and develop models and refine these, ask questions and take nothing at face value. You'll notice that we have one of Clarice's favorite cartoons over here on the right.
Oh yeah, I love that one.
Do you want to go through that?
All
briefly?
Farsight is great. Well, they can read it, but basically, these are some scientists who are, awfully smart. They sure know how air in caves works, but they're kind of disregarding some pretty obvious evidence.
There's some
right in their
red
face.
there's some red flags here. You got
red
a garbage
flags.
can full of Explorer.
One of them
hats and
seems to
bones.
notice. The other one is, he's all about the
red flag.
whatever he's going to learn in that cave, which
But he really wants to get into the
will be
cave.
short-lived.
He really wants to go into the cave.
He sure
right?
does.
He knows a lot about
Mysterious.
caves,
Yeah,
but he's going to ignore some red flags that say,
yeah.
wow, this may not be a great idea. Or maybe I don't understand this particular cave. Maybe I understand other caves. This cave, well, you're gonna add. So we ask questions, take nothing at face value. I've seen caves before, but you know what? Maybe I'm still gonna question this cave.
Maybe this one's different.
Maybe this one's different because you know what? Looking around, it's a little different. So you're gonna ask questions, answer questions, shoot holes in experimental designs and data. You might say, what happened to previous explorers? And you might think, maybe I will change my hypothesis that
right.
this heavy breathing sound is because it's the wind. Right? You show all your data. You even show negative data.
Especially
right?
negative data.
In fact, an empty trash can might be, you'd be good to see.
Might be concerning too.
Concerning
He's
as
extra
well.
hungry.
Exactly. You say, why is there a trash can? Well, don't worry, it's empty. Good news.
Their only hope is the creature is sated.
Right? And then you change your hypothesis, perhaps it's not a great idea to go into
Some
this
of us
cave.
struggle with that.
All right. So how do you do this? Well, you do it through peer review, right? So basically, you write up, if anyone survives the cave, you might write up your results and send it out, and people can, but you do this is what scientists do all the time. People are like, what are you doing, Jones? You're doing this podcast, you're criticizing people. The only difference is, and this is what makes me dumber than everybody else in the field, and you equally dumb, is we do it publicly.
We do.
And put our name behind it and our face behind it
So
because
stupid.
no one else got the stones to do it. Right?
Catch up with us. If you ever hear of us not showing up to work one day, the podcast suddenly goes silent, you'll know what happened.
Don't worry, I have an envelope and a safe deposit
Yeah,
box
that's right.
and instructions on what to do with it.
They won't get away with it.
They think I'm kidding. Anyway, so Dan was very clear about the risks.
Yes, he was.
He was very clear about the risks. Okay. But you go back but the goal of science, the altruistic, this is the AI where A is not in alternative. Wait, I'm not getting all confused on my A's and my I's. A. To get the answer, get the truth, right? I mean, that's the point, right? You change your hypothesis. Of course, again, as we point out, unless you're Tony Fauci, you tell the truth because you can get away with it. If you're big enough, you can get away with it. Actually, if the truth is bad enough, maybe you do want to hide it. So Tony's no idiot.
Yep.
Yeah, he's no idiot.
Everybody knows what side their bread is buttered on.
He waited till he got that prenup type of thing. What is he got? He got the 12-year presidential pardon in advance because everybody knows what he did and how guilty he is and how he basically whatever. But that's beside the point. So, but the point is, scientifically, he was a good scientist at one time. There was a time when he was a good scientist, yes, and not just a political hack. So you have peer review, grant review, get the right answer. And so what types of things are you going to ask? Is this therapy safe? Is the safe dose efficacious? Not just is it efficacious. Is there a better way to do this? Or maybe maybe we should move on. Maybe it ain't working. Boy, your investors don't want to hear that. But you know what? Maybe they should, you know. Sometimes things don't work. All right, so getting back to this, all this stuff kind of went into my head when I'm seeing all these emails, all this stuff. I'm just thinking, oh my God, are there any scientists at this meeting? You know, did people ask questions? Actually, I did hear that there were some good questions asked.
Oh, that's good.
You know, some good people, some good scientists did actually have similar questions to what we have of some of this data at the thing. They just don't want to be very public about it. But that's okay. But the other thing that went through my mind when I was looking at this is this. This is immediately what popped into my head.
So is Red still thinking about giving you the car, maybe?
Even if we do get it, we're gonna need some serious gas money because the cruiser's a boat.
I know it's a boat. This whole gas shortage bites.
Who's getting a boat?
There is no gas shortage, man. It's all fake. The oil companies control everything. Like there's this guy who invented this car that runs on water, man. It's got a fiberglass air-cooled engine and it runs on water.
So it is a boat.
No, it's a car. Only you put water in the gas tank instead of gas. And it runs on water, man!
I never heard of this car.
All right, so I just picture that must have been kind of the scientific discussion around this stuff because, you know, it's too good to be true, right?
Yeah.
Oh my God, it's a car that runs on water, man. Anyway, so Epicrispr reports the first clinical evidence of increased lean muscle volume in patients with FSHD following treatment with Epi321. Okay, and so this was a press release that came out at 8 a.m. Eastern time, and man, everything just kind of went great. So let's, now we've seen their preclinical data, and we've had several podcasts about it. So let's just you know, review briefly. So first off, we love CRISPR inhibition, right? That's why you're the CRISPR goddess, right?
Yeah, we're doing it.
Differently.
Yeah.
Okay, and as people know, conflict of interest, whatever, you might want to say that we have a bias. We do have a company, Renogenics, where we're doing our own version of CRISPR inhibition that we, Carice invented back 2015. And we've been progressing it. We are of the opinion that slow and steady wins the race to answer questions in the lab before you put them into people, so that whatever goes into patients has the best chance of success.
Yep.
And so when we see red flags, we don't go into the cave, which is why our hats are not in the trash can, where some other people are likely to be in a couple of years. So, all right, so we're very supportive. So we did have some concerns, mainly that the target tissue is mature skeletal muscle, and it's not clear that you can ever even methylate non-replicative genomes. And if so, even if you were to somehow get methylation of DNA, because replication and methylation are coupled, but you can imagine if you that you can use bacterial methylases and you can methylate a plasmid. In the lab, you can take naked DNA and you put it together. But we're talking about in the context of a genome, nucleosome, chromatin, and everything, putting in a methyltransferase. Can you methylate the DNA? Still hasn't been shown. And will that then establish repression? That hasn't been shown. They haven't shown it. Nobody's shown it. Yep. Every experiment that's ever been done is on replicating cells.
Yep.
Okay. So the preliminary data test was not very convincing. And honestly, I don't know if they showed methylation or not. It seems they have a methylation assay that they claim shows an 8% increase in methylation.
We couldn't figure out how they analyzed it.
They have a promoter that drives expression of their CRISPR machine that does not, that we know what it's called, right? It was CK8. A lot of people use this in CK8E, right? CK8, CK8E. Doesn't really matter. It's a well-established promoter. that's really specific for skeletal muscle and heart. I mean, striated muscle.
Striated
right?
muscle, yeah.
And boy, it sure was active in a lot of different tissues that's not normally active in.
It didn't look like CK8 or CK80, I'll tell you that.
Looks like they
whatever
got-
they were using, yeah.
They should have sequenced it.
I don't think it was, but anyway.
Anyway, it's very unusual, but whatever. We'll take them for what their data just, the data's the data, what it is. And, okay, the preclinical data that they showed for dosing, in my opinion, suggested that they needed a high dose.
Yep.
Yet they claim that it supports a very low dose. Very confusing to me. They used RH74, AAV RH74, which is in the clinic, and the rationale is it's in the clinic, and a lot of people are serotype negative for RH74 neutralizing antibodies. Other AAVs might have more, but they actually, it's really poor neuromuscular disease delivery vehicle.
Yep.
Right? But it has a pretty good safety profile at low doses. And they're using a shockingly low dose, in my opinion.
Yeah.
There are much better myotropic AAVs available and being used in the clinic, and they chose not to do that.
Yep.
Right? So they're cheap. The non-human primate data indicated they would need triple immune suppression. Okay. Their first cohort is single immune suppression that their own data said wouldn't work very well. Now, what they're going to say is it doesn't work very well, and look at our spectacular results. Well, we'll see about that.
Yeah.
Okay. They did a very short analysis time in their NHP data, and it suggested that their Cas protein is likely going to be cleared over time. Now, we actually saw in our own project that the Cas protein can be cleared over time.
Yeah, in large animals for
We're
sure.
talking about a lifetime, a couple $1 million for a lifetime treatment. Better stick around. The data that we have with a very different Cas in a different system in the pig model suggests that you got to do some extra tricks to keep durability. And our prediction is that overall it's likely safe, but ineffective.
Yep.
Well, we were clearly wrong because those press releases and the coverage from the foundations, the unbiased foundations, remarkably positive. So what was in the news? So, Fierce Biotech, Epicrispr gene silencer's first drug to boost muscle in form of muscular dystrophy, biotech says. It's interesting that the quote, I love the way it's written. They could have left it off where biotech says, but they basically denied all culpability here because they're not saying we didn't say this because we have no idea if this is true.
Yep.
But the biotech says this is true. So first drug to boost muscle in form of muscular dystrophy. Inside Precision Medicine, pump you up. You remember this from Saturday Night Live? Hans and Franz.
Yeah.
We are here to pump you up. Hans and Franz, check it out. I'm Hans, she is Franz, and we are here to pump you up. And that is what this is doing. Epigenetic editor drives muscle growth in FSHD patients. Saturday Night
It's a
Live.
good thing we don't drink before we do these.
Saturday Night Live used to be funny. Seriously, folks. It really did.
Yeah.
Go check it out in the 80s. Everything was better in the 80s and 90s. All right. So let's read more about this report from Fierce Biotech. Other attempts to develop medicines in FSHD have struggled, most notably Sanofi's licensed program from Fulcrum Therapeutics. That candidate, lesmapimod, failed spectacularly in phase 3 trials back in 2024.
Yet people were just as excited when the press release.
Well, that's right. The phase 1/2 on lesmapimod said
forget.
better than expected. The FSHD Society was falling over themselves about how amazing it was.
So really, it's best to take these things with a grain of salt.
Interestingly, the patients that were in the trial were telling me that it was life-changing. It's amazing how good they feel. It's of course, they didn't even know if they were on placebo or not.
Yeah.
But a very similar type of thing. And they actually had better data. But anyway, the interesting thing, though, is from this first sentence, the first 2 sentences, is it still wasn't a failure for Fulcrum. They were able to sell this crap to Sanofi.
Yeah. So we have to keep that in mind.
So they're geniuses.
Exactly.
They sold their magic beans that were not so magic to Sanofi. It's just a bunch of idiots.
This is going to be a recurring theme.
The current theme is big companies and big financial people evidently need better scientists
They do just
advising
fine.
them. And it doesn't matter.
Yeah.
Just like with CHIP and Solve FSHD, $100 million down the drain doesn't matter. So whatever. It doesn't matter to Santa Fe. Whatever. But Fulcrum made a killing. It's great. All right, continuing. Roche's Genentech 2 recently scrapped a phase 2 FSHD trial. I give them a lot of credit for this, actually, for its anti-myostatin antibody, because it did not consistently deliver the hoped-for improvements in muscle growth and function.
That's what you want to see when something doesn't work.
So it doesn't work,
Not to
and they
continue
chose
on.
not to continue.
Right.
You know what? I have a lot of respect for Roche, Genentech, you guys. Good job. Not good. I mean, sad that it didn't work, but you know what? I'm glad you're not just saying, hey, you know what? We can somehow monetize this by going to phase 3.
Yeah.
Great job. Way to be scientists and proper stand-up pharma. They're big enough to be pharma. Friend Eric always he's like, yeah, you got to get pharma and biotech right. It's different.
Yeah.
Anyway, I believe they're pharma. Others racing to become the first with an FSHD drug are Sarepta Therapeutics with an SI candidate licensed from Arrowhead Therapeutics, and Novartis, which acquired an RNA contender of its own in its $12 billion buy of Avidity Biosciences. All right. So when it comes to the competition, though, Epicrispr is clear on where it stands. No other drug candidate has ever shown the ability to boost muscle in patients with FSHD, the company said in its release. Again, we didn't say it. The company's saying it. Interestingly, these guys actually did a little homework. A 2025 study from the University of Rochester, though, found that 19 men with FSHD treated with a combination of testosterone and growth hormone gained an average of 4.5 pounds of lean muscle.
So what do you know? There are ways to do this outside of DUX4 targeting therapies.
Wow, yeah, because those people are still expressing DUX4. They got a little testosterone, a little growth hormone. 12 weeks, I think, is what the study was, right? Wasn't it that long of a study? Maybe about 24 weeks even. And they put on a heck of a lot more muscle while DUX4 is still going. Huh. So
interesting.
So you better know whether or not DUX4 is turned on or off if you're going to claim that your drug is doing something
Especially
special.
if you're treating with something other than just the DUX4 targeting
Well,
therapy.
now wait a minute. I don't see anything. All I see is Epi321.
Well,
I haven't seen anything
let's
else.
look at the data.
Okay, because maybe I didn't look hard enough. Okay, we're gonna pump you up with epigenetic editor drives muscle growth in FSHD patients. Unlike conventional gene editing therapies that permanently alter DNA sequences, Epicrispr uses their gene expression modulation system, and I got nothing against this system. Okay, it's called GEMS. Epicrispr 321 is an AAV-delivered epigenetic gene therapy that restores DNA methylation and suppresses DUX4 expression. Okay,
Really?
so this story, based on their press release after talking with them, says this drug, this construct, methylates the D4Z4 and suppresses DUX4.
Well, I guess they showed it once in their preclinical data, so that's good enough.
This is all about their. They must be showing this.
You would think.
It's inconceivable.
And that word does mean what we think it means.
Thank you. Okay, what does Amber say? We don't cut DNA, that's for chumps, right? I agree with you, Amber. We're not using Cas9 in the traditional editing sense. We use a dead Cas protein, an epigenetic mechanism to silence the gene. Well, again, I can't wait to see the data that's what you're doing. According to Epicrispr, preclinical studies demonstrated that the epigenetic modifications persist through many rounds of cell division, suggesting long-term suppression may be possible after a single treatment. Now, I gotta tell you, this is coming from...
They know what their target tissue is.
Do you guys have any idea what you're talking about? I mean, seriously, it's one thing to misspeak. It's another just to...
This is embarrassingly
It is embarrassingly
ignorant.
stupid.
Yes.
If you are in your
in the
preliminary
field of
exam
Muscle.
in a PhD program,
Muscle gene
and you
therapy.
make this statement, you fail.
You fail, yeah. That's right. And your committee laughs at you
and your committee laughs at you, and
eats more donuts.
I mean, this is so patently stupid. Or is it ignorant or stupid? I don't know. What's the right word on this?
In this case, it's stupid to be ignorant because they should not be ignorant.
But at this
They're
point,
running this trial.
At this point, you're in a clinical trial
With real people.
and you're bragging that this is going to go through. You're in a tissue that doesn't replicate.
So tell us about skeletal muscle.
Tell us a little bit about skeletal muscle. Okay, well, first off, the promoter. If it's functioning
if it truly
properly,
is the CK8 promoter.
shouldn't really actually even be on in replicating cells.
No.
Although, when in the context of a gene therapy vector, it could be. We see it in mud, so they had it all over the place. But it's an interesting thing. They're basically admitting that their preclinical studies were in replicating cells, which is what we have always said. So So they're telling the truth. Their preclinical studies do show that they can methylate and silence replicating cells. Unfortunately, that's got absolutely nothing to do with a muscular dystrophy patient with FSHD who is getting this drug, who has skeletal muscle that is not replicating.
It is post-mitotic. It is quiescent. It doesn't divide. You know, the only cells in your muscle that are capable of dividing muscle cells are the satellite cells, which are the muscle stem cells. They have a specific niche at the sarcolemma, which is the muscle plasma membrane. And when you injure your muscle or you exercise, which is a type of injury, those cells get stimulated to actually divide and
this
form
promoter is not on
myoblasts,
those
which are-
cells.
That's right.
There's no way in hell this is on the satellite cells.
No, people don't have a satellite. The satellite cells are difficult to study because as soon as you take them out of the muscle, they're not really satellite cells anymore. They're kind of activated myoblasts.
So now let's step back and we could say, you know what? Maybe this isn't stupid and ignorant. Maybe it's brilliant. Maybe because you're basically completely misleading. Epigenetic people, the whole point of epigenetics is maintaining the epigenetic state through replication. Hey, we're an epigenetic company. We're maintaining it. And just ignore the fact that it is completely irrelevant to the disease and the construct and anything that you're
It's
doing.
like they did a really quick, look through the literature on abstracts. What is this epigenetic stuff? What is
this thing they call
methylation?
epigenetics?
Oh, okay. I think I got my head wrapped around it. It relies on cell division. Okay, well, let's throw that in the press release. That sounds really good.
I mean, come on, man.
Jeez.
Come
All right,
on.
well, anyway.
Anyway, whatever. They're very proud that they're able to establish. But again, I think it does get to the fact that it is true. Their preclinical data does show that they're methylating and replicating cells, which is why it is worthless preclinical data.
Yes.
And doesn't support
Anything.
anything. All right. So we're going to go back to Epicrispr reports. Okay. So assuming that this is, we've seen the stories, does it actually work like they say it does? What does the data actually say? All right. Now remember, Epi 321, just like Amber says, methylates the D4Z4 and shuts off DUX4 expression. Okay. And this is the mechanism of action right here. It's going to in FSHD, the D4Z4 is epigenetically dysregulated. You've lost the methylation and lost repression. It is poised to be expressed, and they are going to add this methyltransferase targeted with a Cas protein to methylate the DNA, which they say will then establish epigenetic repression. And that is how this thing works. And so what does their preclinical pharmacology predict? It says durable suppression through cell division in cells that don't divide, continuous protection against DUX4-mediated muscle toxicity, even though they didn't ever actually look at muscle. They used an immune-suppressed mouse that does not have DUX4 muscle toxicity. They did not, in non-human primates, it does not. In fact, they have not a single piece of data that says muscle. Now, they did do the apoptosis in dividing myoblasts. I guess that's what they're referring to, but no muscle biologist would call that muscle toxicity.
Yeah.
You just call it cell toxicity. And they rescue DUX4 pathology at clinically relevant exposure levels, and I don't know where the hell they found that data either, but again, their idea of clinical relevant and pathology is different than, maybe they ran this through AI. And
Probably.
it's an AI generated
Probably.
slide.
Yeah.
All right, so what's the study going to do? Hey, remember, we're just doing what scientists do. And actually, we do this to ourselves. We do this to other people. Of course, no one in the building will talk to us. I will tell you something.
Well, that's neither here nor
Not
there.
a single graduate student at UNR has
Wants
me
him
on,
on their committee.
has me on their committee. When I got here, I was on a bunch of committees, and very quickly, I was no longer on any more committees. And I have no graduate students either. Nobody wants to be in my lab because the goal is to get a PhD. And they'll give a PhD to a pet rock, evidently. And I think that we're going to put that to the test soon. But you don't.
That's Dr. Rock to you.
This is why I don't let people call me Dr. Jones. because there's no value in the PhD anymore. I digress.
That's a whole
But
other podcast.
that's a whole nother podcast. PhD means nothing. It's a title. You know, who gives a crap? I mean, you know what? It's what you do with your career. You
That's
don't
your
need a
time.
PhD to be good, and a PhD doesn't make you good. And people want to force you to call him doctor, then you pretty much, it's a red flag that they're a moron. Or Dr. Jill Biden. But she's a hell of a doctor, says Whoopi. I guess give Whoopi a degree too. Dr.
Probably
Whoopi,
have. This is how
Dr. Whoopi Goldberg.
celebrities get honorary degrees all the time, and they must just laugh.
That's right. They're astronauts too, right? I think
Yeah.
you go on The View. They went to space, so they're astronauts.
Everything.
Anyway, sorry, we digress. What the hell was I talking about? Anyway,
They're-
no, I was just trying to make the case.
Their original outcome.
I'm
measures.
trying to make the case that we are hard on everybody.
Yeah.
And, you know, when I was at Illinois, everyone had me on their committees because they had good students.
Yeah.
You know, UMass
And
Med,
UW,
good
everyone would have
students.
wanted you on the
UW,
committees,
good students.
absolutely. Yeah.
You know, and I was talking to the chair of our department, Bob. He's not on any committees either. And he said the same thing. He's like, well, I'm too hard. I asked too hard of questions.
Well, luckily, no one from UNR listens to these podcasts.
I know, yeah, exactly. Well, you would think that you want the hardest questions because isn't the goal to get the right answer? We just, AI told us what scientists do. We told you what scientists
And
do.
to learn how to think.
And to learn how to
If you're a
think.
student, that's all you care about is becoming a better critical thinker. That's the whole goal of grad school.
That should be the
it
goal. of
actually
grad school.
isn't, you know, your project. You think your project is the be all and end all, and you know,
But
the world revolves
just
around
like
it.
a pharma
But
biotech,
really, you look back and you think the whole point of grad school is learning how to think.
that's why I tell people it doesn't matter whose lab you go in. It doesn't matter what project you go in. You go where. Now I chose my advisor Jerry Boss because they said you will get the best training as a scientist in Jerry's lab. You will probably not like him. In fact, you might even hate him. Kind of a weird guy, but he is the best scientist at Emory. He gives the best molecular biology, and you will get the best training as a scientist if you go to Jerry's
And you
lab.
did.
So I sucked it up and I went there, and we're not best friends, but you know what? I got the best training. I'm forever grateful to him. And you know what? He ran the lab based on humiliation. You made a mistake, and it was like the army. We're going to tear you down and then build you back up. But man, if you made a mistake, you never lived it down. But you then figured out how to get the right answer. And the answer was, you think about stuff before you do stuff because most mistakes are caused by not thinking about it.
Yeah.
For example, if you said, Jerry, I'm going to test if my DNA methyltransferase gives you repression that persists through replication, he'd say, well, tell me about the cells you're going to work. And I said, well, these are skeletal muscle cells that don't replicate. He'd bat me across the head. You're an idiot. Come back later. I'm watching tennis. I mean, seriously. I mean, they would have, at lab meeting, if I had presented this data at lab meeting, they would have humiliated me to no end and then forced me to go buy pizza and beer for everybody as a punishment.
Rightfully so.
And rightfully so. Anyway.
Yeah, my
times are
training was
different.
just as tough, only without the humiliation.
Well, the humiliation's great. I actually appreciate the humiliation. I need
Steve did all
I
that
love,
without the, yeah,
I love the humiliation.
without the bad stuff.
All right, so first in human study, what are we going to do? So remember, this is phase 1-2. So really the only thing that matters is safety, right?
Right.
In the end, safety.
That is the primary outcome.
If it's safe, you can go to phase 3, and just doesn't matter if it works, especially if your investors are morons, and evidently they are. I mean, we know evidently.
they're still investing.
Oh yeah, Sullivan's all over these. I mean, seriously, they keep pumping money into this. And it's fine. I got nothing. It said, but there's certain things There's a certain level if you say you got to show me this before I'll give them it. This is how Neil Camarda's doing it with the muscle rebuilding. He calls it the salami treatment. Right, and I think that's great. The salami treatment. Now I gotta tell you, this is not a movie from the '70s.
Processed meat analogies.
I would say, don't Google the salami treatment unless it's late at night and you have privacy on your controls on your computer.
You will not get what he's talking about.
But what Neil means is you don't give somebody $10 million up front and then just expect things to go, because this is what happened to the V-Day Therapeutics and other people. Now the other people at Solve don't agree with this, and they just give you a pile of money and whatever. Chips got money, it doesn't matter. And evidently they don't have any scientists that evaluate your program. Well, they do have scientists, but they must have graduated from UNR. Or somewhere else,
so
somewhere
in trouble.
equivalent. Screw it. I'm sorry. It just makes me angry, man. That's why it's taking us a week to get this out. She was so angry when she saw this data.
Oh, yeah.
But again, we're kind of going.
We'll get to the data, we promise.
The point
Stick
is,
around.
the point is of the salami treatment is you say, okay, I like the idea. CRISPR inhibition, love it. Okay, we've seen some of your data. Show me that you can actually methylate and repress in a relevant animal model. There are plenty of models out there that you can do this in. Just do it.
Yep.
Do the right experiment. Do the right experiment, it works, show me the results, we'll go on to the next thing. Right? Doesn't work? Well, go back to the drawing board.
Right.
Great job, Neil. You're more, well, Neil's an engineer. Neil's a scientist. Neil knows what the hell he's doing.
Yeah, he doesn't waste a salami.
He doesn't waste his time. All right, so the second thing they're going to do, secondary outcome measures. They're going to look at vector copy number, this is straight from clinicaltrials.gov, most recent version, updated. They're going to look at cargo transcriptional activity, DUX4 expression, and methylation status. I agree with all of these things.
Absolutely.
Absolutely.
I was excited when they put that up.
This is what you're going to do. I can't wait to see the data. So what did they do? They looked at vector genomes. So they took biopsies, and they looked at vector genomes.
That's about it. What they did with the biopsies.
They did not report transcriptional activity. They did not report target gene expression. They did not report D4G4 DNA methylation. And they came up with new outcome measures that we've never heard of. DUX-Z biomarker, functional muscle test, which we know what those are, and MRI lean muscle volume. Kind of the concept of all that matters is the outcome. If the patient's getting benefit, who cares how they're getting benefit? And that might be valid if you're honest about how you're treating them. This could be a much cheaper treatment than gene therapy, evidently. This could be, the average is, gene therapy is a multi-million-dollar treatment. What if you don't need the gene therapy to get the benefit?
Well, you probably don't if you go down to South America and get some.
Well, there's that too. All right, so let's look at safety. What do we got going here? We have Epi321 demonstrates manageable safety with no serious adverse events in cohorts 1 and 2. That's great. We might, though, say although 100% of the patients did have an adverse event, but that's okay. They went, there's some, what is this, ALT, AST, this is what,
liver enzymes, I think.
Some liver enzymes, which is nothing unexpected.
Expected.
So I would actually concur that,
as we thought, at these doses of RH74, it's not doing you any serious harm.
It's not doing you any serious harm. Okay? Success. Fantastic success. But you know what?
Primary outcome measure met.
But that doesn't get you a press release and stories in Fierce Biotech and every other journal out there.
I could do that with saline.
That's right. Saline is safe. Absolutely. Okay. So now let's see what else they're going to do. This is from their so okay. Epi321, remember, it methylates D4Z4 DNA and shuts off DUX4 expression. That's how it works. Okay. So now this is very different from what they have on clinicaltrials.gov. They're going to look at muscle transduction at 3 months, circulating biomarker 3 months, MRI at 6 months, and strength at 3 months. They have not looking at D4Z4 DNA methylation or DUX4 expression.
Very suspicious.
Why would you do that?
Especially when you have the muscle biopsies.
So you took the muscle biopsies.
If I were a patient, I'd be really pissed, actually, that they took a biopsy and didn't do anything except get vector genome information out of it, honestly.
Well, no, we don't know that they didn't do it. They're just not reporting on it.
That's true.
Okay. So you think we're being hard on them. Let's look at avidity. Avidity, how does Delbrax work? How does Delbrax work?
Well, it targets the DUX4 mRNA. They're trying to actually cleave the RNA, right?
Yep.
And with a DUX4 targeting siRNA. And because you don't have any DUX4 RNA, you don't have any protein. And DUX4 is a protein that binds to its target genes as a transcription factor, turns them on
in muscle.
And in muscle. And so you get reduced expression of the DUX4 targets. And so
okay, so that's how that
less
works.
pathology.
So what do? They show reduced DUX4 target gene expression in muscle biopsies.
Yeah.
And patients treated compared
exactly
to
the data you want to
control.
see.
This is why we're so high on avidity.
Yeah,
And
straightforward.
you say, why aren't you giving avidity a whole bunch of crap? Well,
they did the right experiment.
They did the right experiment and their preclinical data.
So did Fulcrum, actually.
And Fulcrum did the right data, they just ignored the results and their advisors.
The interpretation was a lot, but the data was done. The experiments were done.
The business people took over from the scientists,
Yeah.
and they did whatever, and the politicians that run the thing, who now run the society.
But avidity so far looks great.
So avidity's preclinical data is the same data that they're showing in their clinical trial. It all works. Everything matches up. Looks like it's working. In fact, I'd make the case that even if they're not getting muscle mass back, and even if they're not getting functional improvement, I don't care, because I believe if you get rid of DUX4, one way or another, you will eventually get improvement.
Yes.
And so to me, this is the only data I care about.
Yeah.
Now,
That is
durability.
what honestly matters.
This
That's
is what
exactly
matters.
right.
How much DUX4 you're
Right.
getting rid of, durability, all that comes down to cost and mechanistics of things.
And
But does this work?
And in combination with muscle building therapies that will be coming, this is fantastic.
This is all you need.
You're solving the essential problem.
Right. So now it's just a matter of can we make it better?
Right.
But the technology works.
Yeah.
Great. In patients,
Yeah.
in the right tissue, on clinically relevant, right? That matters.
Yeah.
How about Sarepta? How does Sarepta work?
How does Sarepta work?
Well, it's the same, right?
Oh, you mean their
I'm not asking you how
Sorry.
the company works. I mean, come on, wake up there. She didn't have her coffee. I interrupted her. She told me ahead of time I didn't have my coffee
I answered
today.
the question. You gotta,
How does
words
Sarep-
matter, Pete. Anyway, basically the same thing. The siRNA targeting DUX4.
Thank you.
Yes.
Okay, and what do they show?
What do you know?
The same thing.
Same thing.
Guess what? It
DUX4 target genes go
and
down.
people want to complain, they do it different, they will assay different. Is it 90%? Is it 93? Is it 54? Who cares? It's working.
Yeah. The point is, it's working.
It's an siRNA that targets X4 mRNA that leads to loss of decreased target gene expression. This is in patients. The preclinical data matches with it. The assays you did in preclinical are the same assays you're doing in patients. And everything, and it's working, and you're going to get benefit.
Yep.
And is it going to be better or worse than Avidity? I don't know. Is redosing going to work? I don't know. It doesn't matter. The technology, as the mechanism of action, works. Okay, mechanism of action, Epi321, methylates the DNA, epigenetically silences it, and silences DUX4, and it's not going to actually, so DUX4 target genes are going to go down. How do you determine if it's working?
Well, again, remember that these other companies that do DUX4 siRNA technology, there's a whole mass of preclinical data supporting the mechanism. We knew the mechanism. We knew
We knew
how
it was going to work.
We knew it was
There's no
gonna
way
work.
it's not going to work.
Yeah, and this is completely different. There is no evidence, no convincing evidence that you can
In non-replicating,
remethylate
yeah.
in quiescent skeletal muscle. So already the mechanism of action was very, very sketchy.
So it's not, it's working like it's supposed to, and it works like everyone else gets it to work. It's
Right.
like, we're going to be the first people
We're
to
going
show
to be the
that
first
this
people
works.
to even, because
they
the
seem to be-
preclinical data wasn't there. It just wasn't
Okay,
there.
so how do you determine, we saw how Avidity checks it out. We know how Fulcrum also did, shuts down DUX4, and then Gene and Fulcrum, we could show you the same data. Analyze DNA methylation levels, analyze DUX4 target gene skeletal muscle. Okay, can they do it? Well, this is their own data. Okay, they have a methylation assay that I don't understand at all. They have cells that are very highly methylated that doesn't make any sense. But at the end of the day, they must have taken
did
this
something.
data to the FDA
Right.
and said, we got an 8% increase in methylation, which doesn't make any sense at all. But I'm sure they didn't,
And the FDA's eyes glazed over and they said, run with
whatever, man.
it. Whatever.
I mean, they did an AI off-target screen, which again makes no sense
Yeah.
because the AI off-target is driven by the Cas and the guide and not driven by the protein that's fused to it, which actually is what causes the off-target. But that's, again, FDA is not what it used to be. But whatever.
But they looked at DUX4 expression in these cells.
DUX4, okay.
So they have a DUX4 assay. They have DUX4 target
gene
genes.
assays, apoptosis assay.
So
Okay, so
we
you have all
assumed
the
they did
assays.
this
You
with
can
their
do
patient
the
biopsies.
assays. Okay, let's look. But that was in cells. Here's that idiotic mouse model, or that they didn't do quite right. But anyway, so they used this mouse model that wasn't quite right. But again, the same thing. DUX4 target gene scores. This SLC34 Bob Block's favorite gene here. Things aren't getting repressed nearly as well here. But tunnel score, they don't show methylation, but presumably they did methylation. So they can do it, right? They have the assays in-house. You go to the FDA, you say, as I understand it, here's our preclinical data, here's our plan in our clinical trial. Our preclinical data says we can perform our plan in our clinical trial. And also I've seen so far is that we've now changed it. Now, it's particularly interesting because at the society put out this thing. They said, great start with keynote speaker from Dr. Russ Butterfield looking at D4Z4 methylation in 5 generations of one family. I understand this was a great presentation. Dr. Butterfield, I don't know Russ at all. I tried working with him once. He didn't respond to my email, but again, he's with the East Coast. He's probably not allowed to work with us. But whatever, I understand he does a great job. I understand this was a great presentation. Okay, fantastic. But you also see that he also gave the presentation, Epigenetic Silencing in FSHD, Preclinical Therapy preclinical Epi 321, whatever, is Russ Butterfield. So Dr. Butterfield is one of the world's leading experts on DNA methylation of the D4Z4 and FSHD. He's developed assays and is using assays for his talking for things to assess DNA methylation of FSH of the D4Z4 relevant to the disease. He is evidently advising and working with, I know that Utah's in a site, but considering that he gave the presentation, would imagine he's aware of DNA methylation. The therapy works by methylating D4Z4 DNA and thereby repressing DUX4 gene expression. They took biopsies. They have the biological material. They even have 6 months. They've had 3 months to do it. Certainly, it has occurred to Dr. Butterfield to ask the question, is this methylated? And it certainly has occurred to Epicrispr that if they wanted to know, they know who to call, their main guy. It is inconceivable to me, but my understanding is Dr. Butterfield was asked the question at this and said, what about DNA methylation? And his answer was, do you know?
No, I wasn't there.
He deferred, I'll defer to the company.
Oh, that was wise. And what did they say?
They said nothing.
Well, this is very enlightening.
They said, we have MRI data for baseline at 6 months.
Let's look at our MRI.
Okay, no D4Z4 DNA methylation data, no DUX4 expression data, no DUX4 gene data.
Yeah.
And you got to just be, I mean, at some point you just got to laugh them off the stage, right?
Yeah.
I'm sorry, that's just what you got to do. So, but then again, we are open-minded. I got to thinking, why might they not have presented this? So maybe they just changed their mind.
Well, clearly they.
Maybe they just said, you know what? We don't care if it's methylated. We don't care if DUX4 is turned off. We're not going to bother checking anymore.
Too much
We're not
trouble.
interested if our drug actually works. We're just going to assume it works and look at the downstream things. Okay? Not going to check. Maybe they did analyze it, and it's spectacular. And this is like a rope-a-dope, and they're going to, and surprise everybody. You guys don't think we have the data, but actually we do. Well, I'm glad you asked. It is spectacular. We just don't want to tell you how spectacular it is. And maybe that's the case. I hope that is the case. I'm all for number 2 here. Okay, I'm free for this. Maybe they tried to do it and they can't get the assays to work. Maybe they reached out for help because they can't get things.
Maybe.
Which doesn't make sense because Dr. Butterfield is there. I mean, Russ, show them how to do it. Come on. What are you gonna do? Oh, we got paid. I could direct you to some papers on how to do it. Call my friend Oliver. Preclinical data on DNA methylation target gene expression says, though, that they know how to do it. So it's hard to, I gotta say, it's kind of hard to
wrap our heads around some
had
of
to
these.
say that
Yeah.
we showed we can do it, and now we can't do it.
Yeah.
So they must be able to do it. So either it's number 1 or number 2.
Or
Because the last one is that they actually did the analysis, the results didn't support what their plan, and they don't want to tell anyone. And you know what? They don't have to tell anyone. Now, I don't know what the answer is.
this is perfectly legal.
It is all perfectly legal. If I was an investor with any brains at all,
I would
demand
demand to know.
this data, and before they get any more salami. But if they've already been given the whole salami, you got nothing to stand on.
That's right.
And we know some of their
Let's have 2
investors.
salamis.
And when they come and ask for more salami, I'd be like, you damn well better show them. I hope they have the data, and I hope it's supportive. And I hope that they just come back and laugh. Well, you know what? You didn't show it, and we don't know, so we're stuck giving us with this thing.
I don't think we're going to see anything because what did they do instead of this? They did something instead of this.
Well, they did. But you know what? I'm going to digress a little bit as I do. Because this also put another image in my mind. And these are the things that just pop into my head as I was thinking about
very
this.
ADD in
I
case
walk
you
around
haven't-
and talk to myself.
haven't
Drives Taki
figured
go
this out.
crazy. Drives her nuts. And I talk to myself. She's always like, who are you talking to? Who are you fighting with? Who are you arguing with? And she's exactly right, and that's very annoying.
Demons in his head.
The demons in my head, that is right. The voice in my head is... And so I thought about this. Reservoir Dogs.
Good movie.
Have you seen Reservoir
Yeah,
Dogs?
long time ago.
Long time ago.
Quentin Tarantino.
You don't need proof when you have instinct, is my favorite quote from there. When Joe knows, he knew that there was a rat in the group, you know. But as a scientist, we trust our instinct, but you still got to figure it out. And They figured it out. But the reason I bring this up is because every once in a while, companies and labs and groups, like they do a group photo that kind of is a fun photo, a fun event, something fun that kind of represents the company. And so back in when I was at the University of Illinois, this clown here on the right, John Backus, who is now Dr. John Backus and is an orthopedic surgeon and probably a very good one, even though he looks like an Amish boy going to a barn building here.
He does, with his hammer and his hat.
But he took a photography class, and the assignment in his photography class was to do a photo shoot that resembled the work environment or something about your work. And so this is my lab from the University of Illinois, and he said, "Oh, we're going to do the scene on Reservoir Dogs when they're walking out of the diner after everybody gets their assignments."
You look about the same age as your people.
People used to ask me, "Whose lab are you in?" I'm like, "I'm in mine." You know, I started my lab when I was
Not
31.
anymore.
Not anymore. People say, "I'd like to speak to your son, please."
Yeah.
But anyway, so this is our lab. We're walking out of there. You got Charles looking hard there in the back. You got Steve, and then the other Steve looking there. He's actually a big guy at Sigma. Cheng He has got a lab at UMass Amherst. He's a PI. Fantastic. Probably, you know, there's Ryan there in the back with a piece of wood to do some whooping. Looking hard, looking good. John's going to a barn building. I don't know why. He told us to grab something. I mean, he goes, you know, so anyway. This is us walking out there. I'm clearly the leader there in the front. We did reservoir frogs because we work on Xenophus, African cloud frogs. We thought that was pretty good.
Yeah.
Reservoir
No,
frogs.
that's great.
So it just popped into my head, as it did, and I thought, Epicrispr could do something like this.
They should.
They should do something. What would they do? And I thought the movie Now You See Me, Now You Don't. Unlock the illusion. And I'm like, yeah, you know, it's perfect, man. You got Stanley Chi as like the Morgan Freeman character, the brains behind the whole thing that's kind of off scene.
We didn't do that for you. We could have, but that would really get us in
Well,
trouble.
you know, Amber, she wouldn't be the Isla Fisher character because she's kind of the boss. She'd be more like the Atlas character. So she'd be, you know, Jesse Eisenberg and would be the boss, you know, and you can put, I don't know, you know, Wes would probably be more like the Woody Harrelson character, you know, and I could do this great steam with Now You See. I actually like these movies.
Yeah, yeah.
The first one was pretty good because you didn't know what was coming. Then the second one, they got to improve. You know, it's this wacky sort of you'll never see it coming sort of magical thing, and then it goes. You know, they're basically kind of like thieves that aren't really thieves. They're Robin Hood thieves. I don't know. Third one, this is the third one. I mean, at some point, it's just like the Mission Impossible. At some point, coming up with an impossible mission that they're going to solve gets really freaking hard, right? And so I can't see how they do a fourth, but actually they're very entertaining movies, and I like the catchphrase, unlock the illusion.
Yeah, it's great.
So I'm saying we should. So we're going to do
That's
that
our
now.
job, to
We're going to
unlock the illusion.
unlock the illusion. Okay. So let's look at their study design. So they're going to do 12 participants, 2 different dose levels. Preclinical data indicated prednisone alone would lead to fewer Cas-expressing cells. We predicted they would need sirolimus and rituximab as well to wipe out the B and the T cells. Interestingly, Stephen Tapscott, one of the greatest minds in the FSHD space, in my opinion, has suggested that rituximab alone would be a therapeutic for FSHD. That's immune suppression would give you great therapeutic benefit. Of course, it sucks to be on immune suppression, but that alone, in fact, we're going to do a pig trial on that. It's actually a great idea. Interesting. So what they're going to do is they're going to shift from, they're going to start out with prednisone, and then they're going to shift, and this was not in the original plan. The original plan was just prednisone, but they now, for reasons they don't tell us, have decided to go to the triple immune suppression, which is not surprising. Later, they're going to claim that they have enough vector genomes per diploid genome than they need, even at the low cohort. So it suggests, though, they're not happy with something. It is true, more Cas-expressing cells would be better. So the real question is, how does the immune suppression affect the outcome measures? Because they're not going to look at DUX4 target genes, or DNA methylation.
Yep.
And all of these press releases, all you hear about is Epi321, and no one says anything about immune suppression.
Yeah. They absolutely
It's almost
should.
fraudulent, but I wouldn't throw the word fraud out there because
We're not even sure that they know this.
they may not even be aware of it. So, and then they're going to talk about how this compares to other clinical trials like Fulcrum, Avidity, and Sarepta. And they're going to talk about the MOVE FSHD and the Resolve natural history trials. But none of these have immune suppression.
Yeah.
So who gives a crap?
All right.
All right. Interesting. There is a body of literature out there. And if you get on one of these fancy computer things, you can say, is there anything known about prednisone and FSHD? Pretty good.
Yeah.
1997, Robbie Tawil. And what do you know? Jerry Mendel, Kissel, the gang at Ohio State. Wow, this is like the who's who of muscular dystrophy circa 1997. So they actually did a pilot trial of prednisone and FSHD. And what did they find out? They did 12 weeks, so that's 3 months. So hey, it's the same length of time. They did manual muscle testing, and they looked at creatine kinase excreted, and they looked at the strength. All right. What did they find out? They found out that the manual muscle testing over 12 weeks didn't change. Some people got stronger, some people got weaker, some people didn't change. No significant data, but it's interesting. Some patients on the drug got weaker, some got stronger, some stayed the same. Okay, how about the maximum voluntary isometric contraction testing? I have no idea what that is, but that's fine, because what matters is some people got better, some people got worse, and some people stayed the same. All right. This is particularly interesting to me.
Yes, it is.
Creatine kinase levels basically crashed. Now remember, prednisone is not affecting DUX4, but so serum creatine kinase, what is that?
That is so creatine kinase is a protein that's expressed very highly in skeletal muscle, and when your muscles are dystrophic and they die, the dying cells release it into the bloodstream, and this is actually a very established and robust marker of dystrophy, circulating levels of creatine kinase. So if the muscle is healthier, less of it is dying, you'll see a drop in serum creatine kinase, and that's what they see here.
So in FSHD patients on prednisone, they get healthier muscle as indicated by a serum biomarker that is not related to DUX4 expression levels.
Yeah.
So DUX4 expression levels, they may or may not be going down. They don't actually know, because in 1997, people didn't know about DUX4.
Yeah, it's really just a marker of muscle death here.
But it's okay. But it's important to know that prednisone alone
Can do this.
it can do this. Interesting criticism brought up. One possible criticism of the present study is that we did not select the patients for the presence of inflammatory changes on their biopsy results. They didn't do MRI back then, as such patients may be particularly steroid responsive. Meaning that if you're in the trial and you have a lot of muscle inflammation, you are going to get particular benefit from the prednisone.
Yes, exactly.
If you have very little inflammation, you're
You may
not
get
going to get
less.
as much benefit
That's
from the
right.
trial. So one question might be, right, what's the deal,
Yeah.
right? So what go back to says, however, there's no evidence that the presence of inflammation in the muscle biopsy specimen in patients is predictive of disease activity. That is no longer the case. We know now that that has actually been shown, and there's no evidence that such patients represent a genotypically distinct group of FSHD patients, which basically you get a lot of inflammation in FSHD and it does predict disease.
Right.
Okay. And if that's the case, and you stop the inflammation, that should actually give you benefit.
Yeah.
Would be the logical prediction. I could put that into AI, but I'm not sure what it would say. What would happen if you blocked inflammation in FSHD? We could put that into AI
Oh, yeah.
and see what it says. But in my opinion, the absence of any placebo group, and by placebo, I don't mean a natural history group, I don't mean Fulcrum's data, I mean, in the absence of any actual placebo group, meaning with immune suppression and going through the infusion procedure
True control.
so that they don't know that they're on placebo,
What you can't do here.
is vital to show direct biological activity and target engagement to make any valid inferences on efficacy of the therapeutic modality.
Yes.
Safety, fine. No problems with your safety.
But is your
Although you-
treatment working the way you think it is?
Is your safe dose efficacious? I still don't know. So let's look at some more data. Okay, muscle delivery exceeds target threshold for clinical activity in cohort 1. So they claim that they were trying to get 3 vector genomes per myonucleus.
0.3.
For 10 myonucleus.
Oh yeah,
per
right,
10.
right. 3 per 10. Yeah.
Which is just a weird target. I mean, that's like saying, okay, you know,
Every 10. Three of you are
I'm,
good.
I'm 50 pounds
Or one
over.
of you is
I'm
good.
50 pounds overweight, and my ultimate target is to become 48 pounds overweight. I want to lose 2 pounds, and I will make it. Damn it, success! Because we're going to pump you up. And so the rationale up here. Okay, so they're doing this, but there actually is a scientific reason behind this, which is not as idiotic as replicating cells. It is the idea that people who are mosaic for FSHD are mild to asymptomatic, so you don't need to hit every myonucleus in order to get benefit. I agree with that. That is actually true. However,
Great,
more
you could
would
hit
be,
just the
but more would be
just the
better.
DUX4 ones.
But wouldn't it be? We know from the tremendous work in the Duchenne field,
Yeah.
it's not random throughout. It's very patchy. That you might get a 10 myonuclei here and none over here, and it's not like spread like through a cell culture plate or a 3D system.
Right.
You would know this if you actually looked in an animal model that was particularly relevant. Hypothetically, if they were available,
Yes.
which they are. But anyway, so they got it. Success. This is success. Okay, fine. But interesting that they're going to go for mosaic of FSHD. So what is mosaic? So mosaic FSHD means that the mutation in an individual, the spontaneous mutation, happened in the 2-cell or the 4-cell stage, right? In which case, if it happens in the 2-cell stage, then 50% of your cells are genetically not FSHD and 50% are genetically FSHD. If it happens in the 4-cell stage, then 25% of your cells are FSHD and 75% of your cells are not FSHD. We find this in a lot of parents. So if you have a child with 3 repeats that's very severe, it is often very common to find one of the parents is mosaic for that 3 repeats, especially if there's 2 kids or 3 kids that have that very severe, because it's much more mild. And so this is a rational, makes sense basis to say we don't need to hit every. In fact, we use the same rationale that you don't have to hit every myonucleus. However, more would be better. Okay. So the example here on a biopsy over here on the left, the blue dots are nuclei. Not all of them are myonuclei. Some are blood cell nuclei, some are parasites, some are 5 FAPs. And I don't know how to tell which one's which, so I colored them green. The green ones that I figured were myonuclei are FSHD. DUX4 is on. It has the ability to be on. And when you hit them with their Epicrispr thing, the idea is you're going to have fewer green myonuclei. There's some red ones over there, and these red ones are going to be epigenetically off. And if enough of them are off, you've essentially created mosaic or asymptomatic, and you fix the disease. And you know what? I'm fine with that rationale. More is better, but that's what you're going for. There's nothing scientifically flawed about that, in my mind. I'm fine with that. However, if we look at the data, and what we see is vector genomes per diploid genome. It is well known in the space that this doesn't correlate very well with positive fibers, right? Because when you do this, you get a lot of virus that is not actually inside the cell. A lot of virus that is not actually, so actually the number of positive cells is actually far fewer than what you would predict on cell culture data that doesn't have all of this other problem.
Yeah, the in vivo situation. Yeah.
So we know, however, so we know this from all the dog data, all the non-human primate data. There's tons of data out there on this. We've covered it in previous podcasts. However, their data appears to be built on cell culture data. And what they show in their cell culture data is that if they need 12 VGs per diploid genome in cell culture to get a 45% reduction of DUX4.
Yeah, that's right.
Okay, so they're saying that they have basically 1.2 and they love it. They're loving it, right?
If only we had some DUX4 data. It's hard to make much out of just the vector genomes, guys.
Starting to understand why they chose not to do the analysis instead of hiding it,
Yes.
because choosing not to do it is
I think we can imagine what this graph would look like.
All right, so they're saying that repressed apoptosis, again, 12 vector genomes to get a 40% reduction and a 30% reduction in apoptosis. So again, maybe it's working exactly how they think it's working, except for the replication, and it just ain't that impressive. Except that they got muscle mass, so we haven't gotten to that yet. Okay, the dosing determinations and preclinical work in likely replicating cells and never targeting the true in vivo tissue in their preclinical data, differentiated skeletal muscle, to me, and you can sign on to this if you want,
Yeah, of course.
is highly questionable.
Yes.
All right, you're going to seal your fate. I'm telling
I
you.
have been. What have I been saying all this time?
That I forced, well, people have turned
yeah,
on me in
yeah.
the past. People have turned on and said, "Oh,
Yeah,
I had,
I know.
he's my boss. I gotta do what he says. I
They
gotta do
put
what
me
he
up
says."
in front of the judge.
I know, exactly. Don't worry. The firing squad's just going for me. Don't worry. I take that back. I view myself more as William Wallace.
Yeah.
Okay, and they're going to cut me into quarters and send me to different parts of the kingdom and put me on stakes. And then you're going to be Earl the Bruce, who's going to lead us to freedom.
Okay. They didn't disavow you slowly. I
And
remember
then
that. Yeah,
he
yeah.
didn't fare too well.
Yes, no, he didn't.
But Earl the Bruce then earned to have a great beer named after him by leading Scotland to
That's
freedom.
what matters.
At Falkirk, I believe.
Oh, really? See, these are the tidbits I don't know. It's a full education in the Jones Lab.
That's, anyway, our lab has put a lot of effort into creating the tools to move FSHD therapeutics to the clinic, including the pig, which has been available since November of 2023. You can do the experiments we're requesting. Other people are doing, we're doing them. Lots of people are doing them. You can get these answers if you want to. So we're not being too crazy.
No, not unreasonable.
Okay. All right, we're going to break for a song, which I haven't decided on yet. And you won't get to know what it is because I'll put it in post-production. If you want to know what it is, you got to get invited on a more professional podcast. But we'll be back in just a moment.
Oh, joy.
Harry Truman, Doris Day, Red China, Johnny Ray, South Pacific, Walter Winchell, Joe DiMaggio, Joe McCarthy, Richard Nixon, Studebaker, television, North Korea, South Korea, Marilyn Monroe. Rosenberg, Sage Bomb, Sugar Ray, Panmure, John Randall, the King and I, and the Catcher in the Rye, Eisenhower vaccine, England's got a new queen, Marciano, Liberace, Santayana, goodbye. We didn't start the fire, it was always burning since the world's been turning. We didn't start the fire, no we didn't light it, but we tried to fight it. Joseph Stalin, Alan Cobb, Nasser and Prokofiev, Rockefeller, Campanella, Communist Bloc. Roy Cohn, one for all, Toscanini, Dacron, Gen, Food Falls, rock around the clock. Einstein, James Dean, Brooklyn's got a winning team. Davy Crockett, Peter Pan, Elvis Presley, Disneyland, Bardo, Budapest, Alabama, Khrushchev, Princess Grace, Peyton Place, trouble in the Suez. We didn't start the fire, it was always burning since the world's been turning. We didn't start the fire, no we didn't light it, but we tried to fight it. Little Rock, Pasternak, Mickey Mantle, Kerouac, Sputnik, Joe and I, Rig on the River Kwai, Lebanon, Charles de Gaulle, California baseball star, with the homicide, children of the little mine. Buddy Holly, Ben Hur, Space Monkey, Mafia, Hula Hoops, Castro, Edsel a no-go. U2, Simon Reed, Paola and Kennedy, Chubby Checker, Psycho, Belgians and the Congo. We didn't start the fire, it was always burning since the world's been turning. We didn't start the fire, no we didn't light it, but we tried to fight it. Hemingway, Eichmann, stranger in a strange land, Dylan, Berlin, Bay of and martial law. Rock and roll and color wars. I can't take it anymore. We didn't start the fire. It was always burning since the world's been turning. We didn't start the fire. But when we were gone, it was still burn on and 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 on. We didn't start the fire. It was always burning since the world's been turning. We didn't start the fire. No, we didn't burn it, but we tried to burn it. We didn't start the fire. It was always burning since the world's been turning. We didn't start the fire. No, we didn't.
All right, we're back after the break. We didn't realize we'd gone on for 70 odd minutes, and we haven't even really gotten to the interesting data.
No.
But we just can't help ourselves. If you're not having fun, right?
What's the point?
Yeah, we have a good time in the lab. But you know what? Honestly, people got to understand, right? We are putting ourselves out there. But the reason is, I think it's important. I mean, people are signing up for clinical trials. You guys are,
Yeah.
we're not trying to be Debbie Downers or whatever they call it, or is that a phrase or whatever? We're
Yeah,
just trying
Doubting
to.
Thomas. And
I'm a doubting Thomas.
Debbie Downer.
Debbie
yeah,
Downers.
whatever.
Maybe I don't know what these phrases are. So we question things. We question everybody. And you know what? When we tell you that something passes our muster, our test,
Our
it's
muster,
because it
yeah.
does. Because we actually treat everyone the same. You see, you're not hammering people, because they do good work.
Yeah, it's just all
I mean,
based
just
on the
come
data.
on, just do good work. I remember someone was asking, I was in one of these meetings, how do you get Jones to like you? I was like, well, just do good work.
Yeah.
We don't like you. We don't
Yeah.
like or dislike anybody. In fact, I look at us as we are Epicrispr's best scientific advisors. Because I don't know who they got, but they need new people. Because seriously, guys, I mean, again, if I will take them at their word. Amber Saltzman has FSHD in the family. Well, some relative has FSHD. I honestly believe that they have the intention of coming with a therapeutic for FSHD. There's no personal ill will.
Yeah.
There's her sister Rachel is the CEO of Scott Harper's company, who oddly enough is one asked them a good question about their dosing, as I understand it, at the meeting, and said your dosing doesn't make any sense. He knows his stuff, man. Scott's a gene therapy guy. He knows his stuff. He's on our side. He's not on our side, but he's on the side of get the right answer and let's get a therapeutic, right?
Yep.
He's got his own gene therapy. He's got tremendous amount of experience. And when someone like Scott Harper says your dosing doesn't make any sense, I don't feel nearly as dumb.
Doesn't make sense.
Let me tell you, it doesn't make sense
Yeah.
because Scott knows what he's talking about.
Yeah.
Okay. And as do, well, actually nobody scientific. Now I will say we're going to get into some data on this cell-free biomarker and this digital twin.
Yep.
And MRI data. And I will say my friends and colleagues are now making fun of me. I was in meetings this week where I asked questions about this, and I got condescension a go-go.
Really?
Oh yeah.
go-go.
A go-go. Like, the lecture, first off, the gall to question some of these people that have developed these essays. And again, we're not knocking the technology, just got a question. I'm always shocked when the scientists are insulted by questions.
Oh, yeah. Not very scientific.
Answer the damn question.
Yeah. Exactly.
We're not saying, and everyone's, you're supposed to ask the question, why are you so awesome? Right. And that's, you want that question, go interview with the society on their podcast. What makes you so awesome and why is your therapy so fantastic? Go there and cheerlead, whatever.
We don't pander to the best of them, sorry.
We're just asking questions.
Yeah.
Like, does your digital twin have prednisone? Do you really think you don't need a placebo? Is this cell-free? Are there other possible ways to improve muscle health and decrease the expression of the cell-free marker that don't affect DUX4 expression? Well, you are the dumbest person in the planet, my friend. I cannot believe I am actually working with somebody who is so incredibly stupid as to ask that question, but I will try to answer it and using small words and things as little as I can, because yes, I know you but I'm going to try to hide the fact that I can't believe how stupid you are to ask that question. Luckily, I have my favorite foil with me.
Foil.
And we
Yeah.
will, we will together
Sounding board.
ask more stupid questions.
We will. Absolutely. Let's get to it.
I love it when people treat me like I'm a moron because you know what? I am a moron. I am the dumbest person in the room and I love it. All right, so here we go. Mentioned that there's a cell-free marker. So we know they don't want to look at DUX4 target genes in muscle biopsies, and they don't want to look at DNA methylation in D4Z4s.
Right.
So what are they going to do? Well, we know avidity has a circulating biomarker, KHDC1L, and that they have validated in patients that we know DUX4 levels are going down,
Exactly.
and they can actually...
KHDC1L mRNA in those muscle biopsies is going down. So it makes sense
So
that the protein...
their drug is actually working on the mechanism of action, is everything is
Right.
working, and they validated this.
Yes.
And
actual
and they're not on
DUX4
immunosuppression,
biomarker.
and so they're not necessarily, okay, so it makes a good case for that.
Yes.
But this is something new because they actually have a patent on that, and not everybody's, in fact, no one's allowed to use it.
Even though the paper's out?
Well, but the actual assay for doing this, and again, whether it's an ELISA or RT-PCR, whatever, it's not freely available yet. Different issue there. Not everybody. Well, I mean, there's a lot of investment in that, and people have patents, and we didn't patent our mice. We don't patent tools for the field because we want to advance therapeutics. Not everybody's on board with that, including the people that are critical of us.
True.
Anyway, so Colorado team identified the circulating biomarker. So what is this? Okay, so it's a cell-free DNA circulating. So what's cell-free DNA?
Well, this is DNA that gets into the blood.
I swear to God, if you said DNA that is free of cells, that would have gotten you a thump on the head.
It's usually in fragments, that gets released and by generally dying cells. And it's really high in cancer functions.
Generally dying cells.
Generally.
Well, because apoptosis,
Yeah.
necrosis,
It can
those
be
are dying.
secreted. It can be
But
secreted
it
too
can
on purpose.
also be secreted.
for real,
Right.
actual biological reasons.
So it can be encapsulated in
vesicles and
in
exosomes
vesicles and stuff.
and yeah.
So Suja Jagannathan at the University of Colorado Anschutz is kind of pioneering some of this with a collaborator who I should know better. But Suja is one of the brightest minds in the FSHD space. She came out of Stephen Tapscott's lab and is doing a great job down there at UC Anschutz Medical Campus. Suja's fantastic. So, it's actually really cool. But so the idea is that DNA is wrapped around nucleosomes to get packaged into the cell. And when cells die, the DNA gets cleaved, and it gets cleaved on the linker DNA that is in between nucleosomes, okay? And actually, when you run DNA on a gel, you can tell that they underwent apoptosis because you get this ladder here on the right, which is very low. I could have found a better picture, but I got shit. I got stuff to do.
Yeah.
I got stuff to do. I can't look all day for pictures and draw
From
pictures.
the
so
80s.
whatever. This is what we used to do. And you see this ladder that is nucleosomal spacing, one nucleosome, and this is indicative of apoptosis. Okay, so Suja actually, this work was funded by Friends of FSH Research, and this is one of the reasons it's great that Friends of FSH Research funds actual research, right? They funded this undergraduate, Stephanie Aramo, in Suja's lab, also working with this Dr. Ramachandran, who I don't know. But they're working on this cell-free DNA as a biomarker. Okay, and now the thing is, this has been going on for quite a while. This actually was worked on even in Dan Miller's lab, Premi Haynes, who is now an advisor for Friends of FSH Research. Amy Campbell is down there at Anschutz. She was actually from Stephen's lab, another outstanding researcher, one of the best in the business. Don't know these other guys, but it's it's not easy. Finding biomarkers and validating biomarkers is not trivial. It's not, it takes a lot of time, a lot of money, a lot of resources. Mm-hmm. Friends of FSH has actually backed this up. And in the end, while we're going to interrogate it, it's actually really good work, okay, if applied to the right situation. So just to be clear, curious about the application of this. All right, so how does it work? So the idea is that in multicellular, I used to teach epigenetics. I know a thing or two about it. Russ and Amber can take my class if they're interested. We do have covered DNA methylation and how you assay it. You know, there's books.
There's books.
You can do AI. Anyway, I suggest checking it out. So what's going on? Well, it's interesting. So in yeast, where there's a lot of epigenetic work, the default state is on because you only have one cell. And generally you need all the genes that you have.
Yeah.
So the default is on.
Right.
In multicellular organisms, where you have many different cells with many different needs from the same genome, the default state is generally off. And so in yeast, you have open chromatin at most promoters, and humans and mice and everybody have closed chromatin, most promoters, until they are activated.
Right.
And so when a gene is turned on, you often get nucleosome eviction, nucleosome clearance, which is very important to do chip assays correct. This is, a lot of people didn't use it to check H3. They thought they were looking at changes and modifications, and they were
geez.
just looking at
They
nucleosome
didn't,
clearance.
compare.
Yeah, people, a lot of,
Yeah.
you gotta do the right experiments, folks. So you get nucleosome clearance, and now you have more exposed DNA, presumably, although it can be bound by polymerase, but available to be cut with nucleases when a cell dies. So when a cell dies, you're going to preferentially cut the active genes
Right.
easily over the inactive genes. So you get a little snapshot of the activation state. And this is the general idea, is that they're mapping nucleosome positioning, correlating with activity. Now the devil's in the details, so I hear. I've heard. Because nucleosome, and they now can do these studies a little bit better. And while it does true that TSS, a transcriptional start site, on high expressed genes is often cleared, on other genes, you actually still have nucleosomes there. And in fact, DUX4 target genes, I believe, just recent work shown, activates nucleosomally occupied promoters, but not always, evidently. So it's not a total snapshot, because as we're learning more and more, you may or may not have clearance at the promoter. Okay. So what's going on? What's going on here? So what the team at Colorado did, as I understand it, and I haven't seen the paper or the presentation, but I'm just my interpretation, is that they screened the cell-free DNA, said, what do we find? They may have specifically looked for DUX4 target genes. That makes sense. That's the type of thing you might want to do. Or just looked at all the DNA and say what's there. But anyway, they got cell-free DNA. You could get this from cells in culture that are being forced DUX4 expression and start to find what you're doing, and then move to serum in patients to see what's going on. You could imagine a lot of ways that this might actually work. And so basically what they found is that ultimately, when looking in the serum of unaffected individuals versus affected, they found this particular DUX-Z marker is significantly higher in circulating, the cell-free circulating fragment. And this is this gene called ZBTB17, and it is a direct transcriptional target of DUX4. Okay, so that's pretty good, sort of, right? It goes from a level of -1 to a level of +1. So twice as much, I guess, would be the math, in affected versus unaffected, right?
Yep.
And then if you look at severity, it goes from -1 to +1, but it kind of goes up to more the mild and mid and severe. So it goes up with clinical severity. Looks pretty, a lot of overlap with the mid and the severe, but that's often the case because a lot of times in very severe cases, you don't have a lot of muscle.
Right.
And so actually DUX4, now that they don't show DUX4 tracking here, but I'm willing to bet that's in their paper. Suja is very good. Seems like their student's fantastic. So I'm willing to bet this is all solid. I don't have any real problem with saying this data is accurate. So then they're going to say that let's look at Epi321 treatment. Again, Epi321 treatment suggests that the only biologically active component is their gene therapy. Don't want to draw attention to anything. And what do they find? Well, they said, well, you know what? We're not going to look at, even though we took biopsies from these patients And even though we process them for
Yes.
genome, for DNA, and by the way, you can get DNA and RNA from the same sample. So if they have the DNA, they could check DNA methylation. Maybe they did, maybe they didn't, who knows? They have the RNA, presumably, they are aware. If they're not aware, you can use Trizol and you can get DNA and RNA. You know, in the Wellstone, look at the paper, we have this BILAT study where we took biopsies, and actually, Stephen Tapscott in his lab did the RNA from the biopsies, and then they sent us the organic phase of the Trizol, just literally sent us the organic phase, and then we extracted the DNA out of that, and we did methylation analysis of the D4Z4P. So actually, it's published. How to
You can get a lot
do
of
this.
information from the same biopsy.
But it's published online. Actually, these are papers. It's paper, the BILAT study from the Wellstone group. I can't remember. It's Wong. I think Wong's the first author. Wong or Wang, and they're two different people. But it's done. You can take a biopsy and get RNA and gene expression data following this sort of track here, and DNA methylation data.
Yep.
It does exist. The technology exists. Okay. But instead of doing that, they said, okay, we're going to look at we're going to look at the serum. And they looked and they see a change from 1 to 0. So after 3 months of treatment with Epi, okay, it goes down. So this makes a couple assumptions. And again, I got no problem with the assay. Not knocking the researchers at Colorado. Not knocking it, just saying, okay. So presumably, DUX4 is on, activates DUX4 target genes in the same nucleus where it gets turned on. It possibly activates DUX4 and DUX4 target genes in other adjoined nearby. It can move, although they don't move too much. In cell culture, they move a lot, but you tend to have myonuclear and localized environments. Presumably, DUX4 positive muscle fibers will die, but not always. We know that they can turn off and the fibers survive. So the question would be, how many DUX4 positive myonuclei do you need to kill a fiber? And does the whole fiber die? We know that DUX4 bursts. So the question would be, do cells die while DUX4 protein is still present, or sometime afterwards, or sometimes long afterwards? Data shows the initial burst of DUX4 does not kill cells, but imprints DUX4 promoters. Great work out of his Tapscott lab. I think this is some of the most fascinating stuff. The H3X and H3Y. These cells are on, and the promoters are now marked to be turned on better later. So you've had DUX4 on, and the cells haven't necessarily died. Okay.
Right.
So if you had something that kept them in that pre-DUX4, in that state before bursting, something kept them in, as Stephen likes to call it, the simmering state or the percolating state, or that smoldering. That's the word he uses, smoldering, like Nadia Rosenthal.
Racy. Yeah.
Smoldering.
Primed to express on a second.
I thought you were going to say prime, not
Oh,
good.
no.
Smoldering, I don't know how we got there. Smoldering state of DUX4, if you had a treatment, like an immunosuppressant, that kept you in the smoldering state, you might actually think that you've shut down DUX4 when you really haven't. It's possible.
Yeah.
Hypothetically. So, if DUX4 positive cells die, target gene promoter signatures will be in the circulating DNA. Shut off DUX4, the cells don't die. There would be less cell-free DNA in general. So the cell-free DNA goes down. So how you normalize this matters, as opposed to equivalent cell-free DNA from the same source. So essentially, if you stop a massive form of cell death for whatever reason, it could be because of DUX4. It could be, but anyway, so levels are going to go down. But you could conceptually say that the cell-free DNA markers, and this is where I was reprimanded, is a marker of cell death or muscle cell health, like CK.
Exactly.
And because, as I understand it, it's possible to improve muscle health without reducing DUX4 levels. And now, granted, the signature should only be there if DUX4 is on. But again, we also know that DUX4 puts a signature on cells, And I think there's just a lot of unanswered questions.
Yeah, I mean, one I would have if I were reviewing the paper, which we assume will come out, about
we're not reviewing
DUXV
it.
as a biomarker. So far, it's not even in the literature. But one question I would have as a reviewer is, okay, if this is really what's happening, and this is truly a DUX4 biomarker. Other DUX4 regulated genes, there's a lot of them. It's not like this is the only DUX4 target. There are a lot, and they would all be active. They would all presumably be cfDNA in the circulation. Is this the only thing you see? Well, how much of the DUX4 signature is actually represented?
So I actually kind of asked this. So one of the things about it is the DUX4 signature are an embryonic set of genes, right? So presumably the background should be 0.
But they're not just embryonic.
Well, that's now I'll tell you. I maybe I was misinformed, but I actually asked this question and said, and I was basically said that essentially they're only embryonic genes, and so they are only on in muscles that are expressing DUX4. And so if they, there's, and so
I think
is
that's
this
a gross
gene?
oversimplification.
Well, it's okay. So
There are a lot of immune mediators that are DUX4 targets. Stephen Tapstad's done some great work on this.
I understand that. Yeah, there's
It's
a
all
lot
in
of
his paper.
target genes that are immune mediated.
Yeah, immune
exactly.
mediators, there's retro-
there's feedback and there's all
Yeah,
sorts of things going on. Yeah, it's not as
just
straightforward.
embryonic genes.
You'd think that would be clear. Okay, so this is a new outcome measure in lieu. How do you say that? Lieu yeah. Of DUX4 and target expression and D4Z4 methylation levels. We're going to do DUXZ in blood, and it's decreased at 3 months. I believe it's decreased at 3 months.
Yeah.
I'm not going to. I believe that you can draw a line with a black box. So Fulcrum had the same thing. Fulcrum had this data where they said Lasmapimod shuts down P38, and P38 inhibition leads to DUX4 repression. But there's a black box on how that got there.
All right, still there.
And then even downstream of DUX4, it turns out there's a P38. There are a lot of black boxes. Who gives a shit? Let's just go. Right? We got an IPO coming up. Let's just go. What they say? They said we're on rails to the clinic. So shut up, Jones and Hameda. So, circulating DNA fragment corresponds to the ZBTB17 gene. It's a direct DUX4 target. It's elevated in FSHD and unaffected and correlates with clinical variant. I'm good with all of that. Epicrispr claims reduced levels indicate suppression of the DUX4
That's what has
pathway.
not been at all shown.
That has
Not
actually
at
not
all.
been shown.
Not at all.
Now remember, in cell culture, it's different. In cell culture, where you have one type of cell, and that's it, you can maybe show
In
that
that
that's
cell.
the case in that cell,
They haven't
in
even
that system.
done that here, but yeah.
No, but I'm presumably that's the work that
Yeah.
that was funded.
Right.
The foundational work likely
We
came out
assume
of
it
cell
starts
culture.
in cell culture, yeah.
I'm assuming it started in cell culture and then moved to this stuff.
Right.
Okay. Many DNA fragments are released into the blood from dying cells, such as serum CK.
Exactly. That is an
From
established
every type
marker.
of dying cells. Okay,
Right.
it's a marker of dystrophy. Okay, so if DUXZ is a true DUX4 biomarker,
Yes.
DUX4 signature and muscle biopsy should also be suppressed. So is it?
We
Make
don't
the
know.
connection.
Yeah.
Fill in the black box.
They have not done it.
They have the muscle biopsies. I guess they didn't check.
Because it is possible that the DUX4 signature in the muscle biopsies is actually not suppressed, that their drug isn't working, and yet these DUXZ levels could be down in the circulation. I can think of a way that could happen.
They're heresy.
I know,
Heresy.
it's awful. They
they're
burn me
gonna-
at the stake.
well,
However,
actually, I believe you know why they burn witches. Because so we can flip the logic. This is the logic that's going on here.
I float. See who gets that.
She turned me
If
into a
anyone's still
newt.
listening.
She turned me into a newt. Well, I got better. This is my British accent. I got better.
That's pretty good. Better than mine.
That's right. You float, so you're made of wood, so you can be burned. Okay. All right. So we're sticking with the train of logic going here. So where's ZBT B17 expressed normally? From my understanding, from my conversations, it's only expressed in embryos. So because ZBT is a DUX4 target gene, it must only be expressed in the embryo program and in DUX4 positive muscle cells, right?
No.
Okay. Could ZBT active gene structure be responsive to prednisone?
Yes,
Well, that's
yes.
a totally separate thing. So that's wrong, and we don't know about this other one. So, I'm going to make this statement. I'll tell you what, maybe you should make this statement.
Well, yeah, I was looking all this stuff up. Let's
Okay,
see.
you're the one who did
Indirect
all the work.
measure.
You make the statement. Okay, go ahead.
Indirect measure of DUX4 activity. But I haven't
Speak
read
up.
your slides, so it correctly presumes all muscle health in FSHD is due to DUX4 levels. Yes, that's right. Okay, and all serum ZBTB17 DNA is from damaged muscle. That's another assumption. And that it would not be responsive to prednisone or other immune suppression, for example, sirolimus or rituximab, and we know that that's another assumption that is patently
So,
false.
I think that these are all called into question.
Oh yeah, at the very least. Let's
Okay.
talk about why.
Objection, Your Honor. Can we clarify to the court whether the witness is stating opinion or fact?
This is your opinion?
It's a fact. I find it hard to believe that this kind of information could be ascertained simply by looking at a picture.
Would you like me to explain?
I would love to hear this.
So would I.
All right. Well, sorry, these are the things that pop into my head when I'm thinking about this. I just always pop into movies. This is why I'm a nightmare to live with. Okay. Let's do some digging.
Yeah, so what is ZBTB17, this protein that happens to be a DUX4 target? Well, it is also a transcription factor, and it's actually critical for the development of lymphocytes, which happen to be in the immune system. Yeah, immune
Lymphocytes?
cells that circulate in the blood.
That
you
sounds
know?
interesting. Maybe we'll highlight that.
Blood cells. It works with a very powerful protein called c-MYC to activate its target genes. And yeah, it's actually present. We'll get to
So
that.
this
but a
sounds
lot
like
of
it could be.
tissues.
It could be. I was
Not
gonna say it
just-
sounds like it could be present in a lot of tissues.
A lot of tissues.
It's not
It's
just a
certainly not
DUX4
just-
target gene.
Yeah, it is not just a DUX4 target gene. So it does.
Well, color me surprised.
And then you know what? It functions in adults. So that's how you know it's not just a DUX4 target gene.
I did not see that coming. So, what do we know about
They
lymphocytes?
turn over rapidly,
So wait a minute. So
which means they're dying
They're
in the
dying.
circulation and releasing their cell-free DNA.
So a gene that's
active
extremely
genes.
active in lymphocytes is dying. So this
Developing
could
lymphocytes,
potentially
yeah.
come from.
It could come from the lymphocytes.
So if you
maybe.
had something that
This marker may have absolutely not only nothing to do with DUX4 as it pertains to the disease, but it may also have nothing to do with muscle. It's very
but
possible.
we saw, the data is pretty, it's there's
It correlates.
okay, it correlates.
But what I have a problem with is the study, the actual Epicrispr study.
Well, there's still a number of questions, right?
Yeah, a lot.
It's presented as an okay.
It is
well,
presented as now. If they had shown serum creatine kinase going down, I would have said, yeah, of course it is. That's great. For what it's worth, that's great. And to me, this is no better than serum creatine kinase. It is.
Okay.
They have not shown at all that it's
So
any
as we
different.
discussed, it is expressed in a lot of tissues.
Yeah, look at this.
It's expressed everywhere, including muscle that doesn't have FSHD.
Right, normal muscle.
So actually normal muscle, but that wouldn't be dying. So the argument would be the normal muscle, a healthy muscle wouldn't be dying.
Sure.
And none of these tissues die, evidently. There's no turnover in any of these tissues. But whatever, but that should be normalized, right, among the population. And the special thing about FSHD is the muscle is dying.
Right.
And so there's presumably from the muscle, there's no reason to think lymphocytes, unless there was an immune component of the disease.
Exactly.
Oh, there could be.
Is
I
there?
mean,
Are these patients by any chance
Could-
immunosuppressed?
and is the
And what
immune
would
system highlighted?
prednisone
And
do?
is the immune system particularly active in FSHD? Well, that would be a cause for concern.
I would think so.
That would be a cause for concern. So, you said you'd be curious. Remember, you mentioned creatine kinase.
Right.
So let's go back to that data we saw. We expect CK to go. CK goes down with prednisone because all these patients are on prednisone in addition to Epi 321.
Right.
And we would be curious if you just had prednisone and nothing else, what is the DUX-Z profile? Because if the DUX-Z profile in prednisone alone goes down,
Yeah.
then it's going down independent
Yeah,
of DUX4.
exactly.
Now that doesn't mean it can't be a marker of DUX4,
Right.
but it means there are other ways to manipulate its expression.
Exactly.
Which is important to know.
It is very
if you're going to
important to know.
rely completely on
On
that.
this as the metric of the success.
But they're not
of your-
confined completely
of
on
your
this
treatment.
because they're also doing MRI. And this is really cool stuff. I know FSHD Global Research Foundation is very proud of this. This is from Springbok Analytics and some of the best and brightest Really cool stuff on whole body MRI, Springbok Analytics, Sylvia Blumpner doing this. It's fantastic, interesting, cool work in general. But with Seth Friedman at Seattle Children's, they've come up with this idea of a digital twin, okay? And what they're doing in Springbok MRI is, and many of you have actually had this, if you're in the Move Plus study, if you're in Australia with the clinical passport, you're getting these full body MRIs. And Springbok has this way of they're going to pixelate the fat. Now, let's see, what does that look like? So here's an FSHD patient where they're muscles from the same patient, and they're going to pixelate the fat, and they're really only looking at fat. And blue is 0%, so that presumably that's good muscle. And yellow is going to be 100%, presumably that's all fat. And so you can see over here on the right that you can have the same individual, the pecs can be very fatty with some various spaces, or they can be very unfatty from the same person, right? The other thing to look, and this is, I love this technology. I'm not being at all remotely critical
really
of the
cool.
technology. I absolutely love it. We're doing it for the pig, and Springbok is just the best in the business. I love everything about this. So
Yeah.
just to be clear, in fact, I think it's great technology. In fact, I even love the digital twin, as we're going to talk about it, if it's done right. So here's the glutes. So, glutes. And now you're seeing here, you got some different analysis. Now, when you're going to be using this as a metric to see if you're getting better, if your trapezius over here on the right, how much, what kind of improvement do you need to see here to see, what would 5% improvement be over there? Look over here on the left, what would 5% improvement be, right? It's understanding, it's not just about percent of improvement. You actually want to see what the muscle is like.
Yeah.
In addition, the muscle over here on the right, the trapezius, that is all fat, well, that's not going to be particularly responsive, likely, to a DUX4 therapeutic. I think if you take this muscle here on the right, that's all fat and all yellow, for those of you just listening, and you gave it avidity or arrowhead, it wouldn't do a damn thing because there's no muscle, there's no DUX4 expressing cells.
Yeah, it's just fat cells.
Over here on the left, the glutes, well, you're going to shut it down and you're going to do better. But also over here on the left, you might have a whole lot of inflammation. Okay?
Yep.
And so now you're not just dealing with, it's not just fat. Fat's not the only pathology. Fat is the end pathology. The early pathology on the way to fat.
Well, inflammation and fibrosis.
Now all I can think of is that Kelvin and Hobbes where Susie's there, Susie is in the play on nutrition.
Oh, and he's an onion?
But he said, "What are you?" She says, "I'm fat."
Yeah.
I'm fat.
And she punches him. Anyone else want to say that?
Anyone else want to say? Sorry, I digress. There's more than fat. There's other pathology, and the earlier pathology on the way to fat is inflammation. Let's look at some other muscles. Great variability from the same individual. Tremendous variability in the amount of fat, the amount of muscle. Wouldn't it be great to see the inflammation and see where that is? But you can probably predict what the inflammation. Okay, so what they've done, and this is a great paper to look at, multi-scale machine learning model predicts muscle and functional disease progression. The idea is, let's get away from biopsies. Let's show, the idea is they've taken these full body MRIs and they've gone through these studies, the Wellstone study, the Kennedy Krieger study, Move Plus, Fulcrum, and FSHD Global, and they've tracked people over time with MRI, correlated it with functional studies. And so the idea is, what does the signature at baseline look like? What's the signature going, and can you predict signature, the MRI signature and the functionality, at the end, a year later, 2 years later, whatever. And that's, and so that's the concept of a digital twin, right? The concept of the digital twin is to take your real MRI at baseline and then using AI, so it's good, and say, what would this person be like, and predict what they're like in a year or in 3 months or whatever. And then say, well, now, but they're on the drug, so they are their own control. They are their own internal control, and say they would be like this, but they are like this. And that is essentially what they're doing, this idea of the digital twin. And I absolutely love that concept, because it's AI, so it's good. And I love the looks on the faces I get when they're talking down to me about this. Because I dare to have a question. God forbid there could be a question about this. But the question I have is your digital twin on immune suppression? Is it truly a placebo control? Because the guy that kind of came up and said, you don't really need a placebo. Jones, you moron. And it's great. You don't need a placebo. So obviously, you have a prednisone control for your digital twin. And certainly, you're going to have a rituximab, prednisone, and sirolimus control for your digital twin if you're going to use it in this trial. You must have that in these data, right? Except
they can't.
I'm looking at it. I'm not seeing it, but it's got to be there. I know it's there. It's got to be there. So because it's an untreated digital twin and an Epi 321 dosed is what they say. So the only difference between their digital twin is the Epi 321. Now, dosed is an interesting word because dosed likely includes immunosuppression. You know that.
Yeah, of course.
So what happened? Okay, the first reported MRI increase in FSHD therapeutic context. They had 3 out of 3 participants. They gained 0.8 pounds compared to lose on average compared to digital twins lost 0.6 pounds. Okay, digital twins, they got I don't even know what the hell they're looking at here on the right, but it got better, right? Everything got, that's just looking at the data the same way. Everything got better, right? Increase in lean muscle mass. And it's real because they're comparing it to losmapimod, where losmapimod had a placebo. So for those of you who want us to compare it to a placebo, losmapimod had a placebo, and they did MRI. So and those guys went down.
No immunosuppression.
But the placebo group wasn't on immunosuppression. Just like an Avidity and an Arrowhead and a Souffle, they're not going to be on immunosuppression.
Yeah, this is really
the digital twin is great for these other things.
Yeah.
But if you're going to do it on gene therapy, you damn well better have immunosuppression in your digital twin.
Yeah.
Right? Or
That's what all of that boils down to. Everything in the last 10 minutes boils down to that one sentence. So say it again. If you're going to do gene therapy.
I can't remember what I said. If you're going to use a digital twin, you better have the right actual control for the for the immunosuppression.
And honestly, the data probably doesn't exist for the proper immunosuppression control for this digital twin. I mean, it's all based on a natural history study. They don't have the.
But they do have the data on inflammation.
Yeah, that's true. That would have been better.
In fact, the MRIs are not special MRIs for Springbok. They actually, the way they take the data and post-process it is what's key is Springbok.
Right.
So they should be able to look and see inflammation. Yeah. So what's the data on inflammation? Let's answer these questions. Because I am an idiot. I'm dumb. I want to know about inflammation. Why do I want to know about inflammation? Well, anyway, let's see what they say. They say, as of May 12th, 2026, 9 patients have been treated across 2 doses, with no serious adverse events. That's great. 6 patients were treated, whatever. Among the first 3 evaluable patients, meaning they didn't look at the 3 they didn't want to look at, all demonstrated gains in lean muscle volume at 6 months compared to baseline. Great. Love it. Patients achieved an average of 370 mils of lean muscle volume, equivalent to 0.8 pounds of muscle mass, with individual gains from 0.5 to 1.3, and some muscles gaining 15% in lean muscle volume. But we know that inflammation precedes muscle loss in FSHD. So, in fact, we covered this in the previous podcast. In fact, some of the same people on this Springbok study did this study, where they show that actually fatty replacement comes after, essentially, inflammation.
Yeah.
Estrogen, right? In fact, you can actually see it here. Stir-positive hyperintensity often foreshadows fatty replacement, right? And so you have this data.
Yeah.
So if you're going to say, let's look at fat, so you're saying this is the digital twin, this is at baseline, this is where they're going to be in 6 months. You're going to treat them with an anti-inflammatory. The question I have are the muscles that got better muscles that had high levels of
To
inflammation
begin with.
to
Yeah.
begin with,
Yeah, exactly.
or not,
You would expect
because
so. Yep.
a guy they may have heard of, this guy Robbie Twill, Dr. Twill, one of the great minds of the FSHD space who is now retired, had this paper that said that patients with high inflammatory changes might actually be particularly responsive.
Yep.
Again, without shutting off
DUX4, nothing to do with DUX4.
Now, the answers to all of these questions might be the Epi 321 is working great, but there's
we'll
not a piece of data that shows that.
We will see.
Well, are there other ways to improve muscle health?
Yeah, there's all these
Well,
supplements.
we already read. Well, what do you know? You see, we already have seen in that press release commented, not the press release, the report from Fierce Biotech or whoever said, well, with testosterone and human growth hormone, you can put on 8 pounds or 4
Plenty of ways
pounds
to increase muscle
without
mass.
affecting.
Yeah.
Antioxidants, you can actually improve muscle health.
Yep.
If you go to the FSHD supplements nutrition peer report, you'll see tons of people are improving muscle health without shutting down DUX4.
Yep.
And improving muscle health presumably would lead to a decrease in DUX-Z, a decrease in serum CK,
Yep.
increase in lean muscle mass. In fact, our friend Michael put on 8 pounds of lean muscle mass just by going to the gym.
Yeah.
Without any of this other, well, it did nutrition
Which
and
people
stuff.
do more when they feel good because they've got less inflammation in their muscles.
So let me get this
So there's
straight.
that too.
So you're
There's
telling
no
me
control
that
for that.
when you're in a clinical trial and you got something that makes you feel better, you might actually be more.
You might actually exercise
But they tell
more.
you not to do that.
People don't always listen.
You know, there was a guy that came and visited my house who was in the Lesmapimod trial and was cutting his pills in half and dropping them in water to see if he was on the placebo or not because he was looking for the thing. He's nuts, man. I met a guy in the Fulcrum trial when I was up
Was he
there.
trying to taste the sugar?
You know, whatever, man.
Wow.
That's funny. You ever see Dallas Buyers Club?
No.
Dallas Buyers Club is Matthew McConaughey. It's about HIV. It's actually a great movie. It's a depressing, scary movie. But one of the things about that is they're showing, I think it's very real, that all these people in these clinical trials for the AIDS drugs that are dying of AIDS are actually taking their pills and giving them and selling them and splitting them up, and they weren't actually doing what you're supposed to do.
Yeah.
Well, because they want to live. They want their friends
People
to
are
live.
the worst lab animals.
People are the worst lab animals following the treatment. So they tell you, they tell you to live your lifestyle the same. But I talk to people in these trials and they say, I'm transformed. I can do things that I could never do before.
So they do them.
But you're not supposed to be doing things you could never do before. I'm going hiking. I'm going to the gym. I'm doing great. Well, guess what? All that helps you improve.
It all
Which is
boils
why
down
you need
to
a
the
molecular marker.
Yes, the long-term time point will answer a lot of these questions.
But if you don't ever have a
Because the immunosuppression was only for a few months, so these early
Which is-
time
that's why-
points,
so it'll be curious.
it's not clear what's going on.
But
But by a
again-
year,
But if you've been going to the gym
it's not
for a
working,
year,
it shouldn't be working. Yeah, well, we'll
you
see.
you seeing some improvement, you going to the gym? I mean,
If there's no long-term crash, they're going to monitor these patients for up to 5 years, right?
oh yeah.
so
No, I
we
know.
will see.
There'll be a public company. Everyone will cash out by the time this comes crashing
The
down.
truth always comes out.
So, so I have no problem with this, except it should be this.
Yeah.
Prednisone. In fact, you could make the case that you get this exact same data
Yeah, without
in the
Epi
absence
321 at
of
all.
Epi 321.
Exactly. That is the
Okay.
problem.
Same thing here. Quantitative muscle testing. Well, what do we know? We know that prednisone improved that.
Exactly.
Some people got better, some people stayed
Yeah,
the same,
that's-
some people got worse.
That can all be attributed
What do we
to
know
that.
about the Resolve trial that they're comparing to as a natural history study? Nobody's on prednisone. Nobody's
right.
on immune suppression. They all know they're not in a clinical trial. There is no
Yep.
placebo effect.
It's a very low bar compared to with prednisone.
So we're going to go back here and we're going to look at this measure and say, we know the circulating DNA is critical for lymphocyte development. We know that chronic prednisone decreases circulating lymphocytes, which would decrease the amount of DNA.
Yep.
We know that the cells that are dying can be lymphocytes, they can be muscle, they can
Yep.
be other cells.
Can certainly
And we know that
contribute.
immune suppression will affect this.
Right.
So we're not saying it
These are
doesn't
just
track
concerns.
DUX4. And I'm not saying that this wouldn't be a great marker for other types of trials.
Yeah.
But for this trial, it could be a great marker for this trial. But in the absence of showing a direct effect on the
A
mechanism.
direct connection to DUX4. I don't see how it's any better than serum creatine
So
kinase.
functional tests, okay? Patients underwent immunosuppression for 3 months. Less inflammation, people feel stronger, they exercise more, just like what you were just saying, right? We know this, right?
Yeah.
There are exercise trials going on to do this,
Yeah.
right? The lean muscle volume compared to projected digital twin on natural history and fulcrum, but no immunosuppression.
Yep.
And if they are on immunosuppression, then let us know. And I'm again, we will correct anything that's incorrect.
Yeah.
But
No, we'd
I'm
love to
concerned.
correct anything that's incorrect.
So the findings presented, potential milestone for FSHD. Historically, patients in FSHD clinical trials, including recent phase 3 trials, have experienced progressive muscle loss over time, in contrast to the consistent increase in muscle volume observed across all evaluable patients treated with Epi321 at 6 months. Epicrispr previously reported favorable improvements across multiple strength and functional measures at 3 months in these same patients. Okay? Epi321 is designed to silence DUX4, the genetic driver of FSHD, using the proprietary system, whatever, without altering the underlying DNA sequence. Okay, the single dose therapy is intended to provide long-lasting suppression of DUX4 activity and durable protection against DUX4-driven muscle damage. Not one, this is my words, not one single piece of data shows the DUX4 gene is silenced by Epi321,
Yeah.
in my opinion.
I completely agree.
Okay.
That is absolutely true.
Patients with FSHD face a lifetime progressive loss of muscle that can affect nearly every aspect of daily living, from walking and climbing stairs to maintaining independence, said Russ Butterfield, MD, principal study investigator and associate professor, University of Utah. Historically, we have had very limited ability to alter the course of the disease, although these are early results, and additional follow-up is needed. I appreciate that. The observed changes in lean muscle volume are encouraging and suggest Epi321 may be addressing the underlying drivers of disease in a way that could ultimately translate into meaningful benefit.
Oh, that's a big
God,
one.
if only there was a way to
Figure
that
It
out.
really was.
Oh my God,
Really.
how could we do this?
I don't know. We've been talking about it for
Well, I told you,
hours.
Dr. Butterfield's one of the, and again, I'm not knocking Dr. Butterfield. He's one of the great he's actually fantastic. I mean, honestly, I'm not knocking anybody personally in this, or even anybody. Well, no, Amber's comment was stupid. Okay, I gotta say sorry. Your comment about how great this is on replicating DNA is just idiotic. But outside of that, we're not commenting on anything. But if only there was a way to directly answer this question and determine if it is addressing the underlying drivers. This is where I should have gone with another. I couldn't keep pulling movie quotes, but all I could think of is Bueller.
Bueller? Ferris Bueller?
Bueller. You know, he's like Bueller.
Oh, yeah.
Whatever. Any ideas? How? Oh God, it is such a hard question. I know. I'm gonna throw this out there and see if it sticks, Russ. Dr. Butterfield, how about we assay DNA methylation or DUX4 target gene expression?
We have the power.
Don't you want to know? When someone asks you, have you looked to actually see if the drug that you're promoting works? Wouldn't you rather be able to say, well, yes, we did, and here's the data, instead of, I defer to the company?
I would. I don't know, man.
You know what? When we had negative data on our CAS stuff in my ASGCT talk, you know what I did? I said, you know what? CAS is being degraded, and we're working on it, we're trying to fix it. You know? Because that's what scientists do.
Yeah.
I guess clinicians don't do that.
No, we could have gone to trial 3 years ago if we didn't care.
The consistency of the MRI findings across all evaluable patients. That's a new term, evaluable.
Yeah,
I gotta start doing that.
Valuable.
In my thesis defense, I'm presenting the evaluable data. That'd be like what that recent defense really said. None of the data was significant, but please ignore that. Seriously, that's the kind of defense we had here. None of the data showed significance, but I'm now going to build my thesis upon this. Rubber stamp, pet rock gets a PhD. Okay, sorry. Particularly in a disease where muscle deterioration is expected over time, said Sylvia Blumker, PhD, Chief Scientific Officer, Springbok. And again, I love the Springbok analytics. I love it. I love the digital twin. I just think it needs to be applied appropriately. Okay? Our quantitative whole body MRI and AI-powered muscle analysis enabled objective assessment of changes in lean muscle volume across 140 individual muscles throughout the body. It is great technology. While these are early results, the data highlight the value of advanced imaging. That is absolutely right. In capturing treatment-related changes that may not be apparent through traditional assessment.
That's actually a respectable statement. If I
That
were
is a-
her and I were asked to say something here, that's exactly what I would have said.
Put lipstick on this pig, Dr.
that's-
Blumker, and she did.
Focus on what's positive about the technology and downplay the significance
She's
of the
got
study.
no egg in the
Absolutely.
game on the
Yep,
thing.
yep.
It is, they used our technology.
That
Our
is a-
technology is great. Here it is.
It's a
And
very
she was
smart statement.
Now, the smart thing is treatment-related changes. And the treatment is immunosuppression.
Yes.
In addition to
Exactly. It's absolutely
Epi
true.
321.
Absolutely
So
true.
Sylvia, that is a great
Yep.
statement, honorable statement,
Yep,
proper
yep.
science statement. Your technology is fantastic.
Yep.
Seth, I love your digital twin.
Yep.
Emma, great job funding this all.
That's right.
We're not knocking the technology,
No, not
but
at
the
all.
application, well, it was AI. Maybe they could just ask AI to make a digital twin that's been on immunosuppression. In order to do that, you'd need to know the inflammatory signature. Wouldn't that be great to see the inflammatory signature
Of individual patients.
of these patients that
Yeah.
are
At
here
baseline, 3
and
months,
see how that
6 months.
correlates, because that could indicate
Yep.
if prednisone, the prediction from Robbie Terwell's work,
Yep. Which
is that,
muscles are
and are the
most
muscles that got better?
susceptible?
Because the one thing about those digital twins is some got better,
Mm-hmm.
some stayed the same, and some got worse. They didn't all get better.
right.
Some did get worse, and some did stay the same. Now, some were predicted to stay the same, and some were predicted, but it's,
Yeah.
anyway.
Oh, it's interesting.
So it's interesting. All right, so they're now going to do cohort 2 and change their immunosuppression. Okay, they're going to do prednisone, sirolimus, and rituximab. And now everything is just going to be, I would say on steroids, but I guess that'd be inappropriate. All right, what do we know from their previous work? We know that rituximab, sirolimus, and prednisone actually greatly improve everything. From the virus standpoint, rituximab eliminates the B cells. The body can't produce antibody. Takes 6 to 9 months or longer for B cells to recover. So even if you're only on it for 3 months, it's going to be,
Yep. Takes
it could
quite
take
a while
the whole year, the whole
for the
course of
effects
the
to wear off.
study.
Yeah.
You're essentially going to be on
Yeah.
rituximab.
Yep. Yep.
Sirolimus basically prevents proliferation of B and T cells, immunosuppression. You recover a bit faster, but essentially you're wiping out the T cell and the B cell responses here. And the prednisone is anti-inflammatory. And we know that 3 weeks treatment can take up to 2 years. So essentially, you're going to be on immunosuppression through the entire course. You can't prove that you're not. Even if you're off the immunosuppression, the effects of the immunosuppression are going the entire time.
Yep.
If your readouts are digital twin and cell-free DNA marker, you better find out the effect of the immunosuppression on your digital twin and your cell-free DNA marker. We do know this makes sense. We actually said they should do this anyway, so I got no problem with them doing this, right? But the expectation is that triple immune suppression will lead to increased biodistribution of the cargo throughout muscle, healthier muscle independent of DUX4 levels,
Yep.
and for a longer time.
Yep.
Everything's going to be better than when they report on the second cohort. I guarantee you. Independent of expression of Epi 321, they're going to improve their quantitative muscle, whatever. I don't know if I got the right term. Their DUX-Z is going to go down. Their lean muscle volume is going to go up. In fact, if they got any brains, I guess they'll be taken, if they want to show, they should get people with a lot of inflammation in their muscles. But what they really should do is get people with no inflammation, very minimal inflammation, to show how great this is, because
Yeah.
that actually would minimize the effect of the immunosuppression.
Yeah, it really would.
They should know the level of that. But that's important. If I were an investor, I would want to know that.
Yep.
Unless I gave him the whole salami already, in which case I want 2 salamis back. That's right. And if an investor gave him a salami and wants 3 salamis back, you don't want to know the answers to any of these questions. You want to say, go public as soon as you can.
Yeah.
Okay. How will they distinguish the dose response improvement of immunosuppression and triple immunosuppression versus Epi 321? You got to do that. Will the digital twin account for the effects of the triple immunosuppression? Will the DUX-Z account for the effects of triple? Again, we're not knocking DUX-Z as a marker, but there are questions about it. As dumb as, again, I'm an idiot, and again, I'm sure this will be explained to me again how dumb I am to question this. Will they ever do any experiment to show directly that Epi 321 is actually working as they're supposed to do? All the siRNA companies are doing that,
Yep.
right? We're doing that.
Yeah.
In fact, I don't know anybody that, Fulcrum kind of did that. I mean, they showed that it's blocking P38. So losmapimod was blocking P38.
They looked at DUX4 targets.
I mean, then they looked at DUX4 targets, and it wasn't working. So actually, the Fulcrum, even though there's some black boxes, they actually did it pretty good.
A better job
they did
than these guys are doing. Yeah.
The Fulcrum did it pretty good. You guys are the only ones not doing it, man.
Yeah, there really ought to be a standard.
All right, but you're sitting here and so what's coming? Well, okay, so let's look at this. What do they say? So, successful enrollment. Good job. Well tolerated, manageable safety profile. Thumbs up. Early circulating biomarker data shows reduction of DUX4Z indicating suppression of the DUX4 pathway.
Nope.
Yeah, maybe it does, maybe it doesn't. I'm not convinced.
Not at all.
It could be suppressing the pathway, but there's obvious ways that it's not.
Yeah.
Okay. Early MRI data shows increases in lean muscle volume in all evaluable patient participants in month 6, indicating gain in muscle mass. That is actually true.
It does.
It does indicate gain in muscle strength. And early strength and functional support the potential for EP321 to provide transformative therapy for FSHD.
If you just crossed out EP321 on this entire slide and put prednisone,
then it's thumbs up all around.
Thumbs up all around.
Everybody
Absolutely.
go get yourself some
And all these
prednisone.
statements are true.
First in human study expected to complete enrollment. Great. Glad you're doing. But here's the kicker. And this is where the partnership and the press and everybody going crazy is they are going to start a new phase 2B study in the second half of 2026, which is now. And so they need a bunch of people to sign up for this. And again, we don't tell people what to do. But they're going to look at other circulating biomarkers. In fact, they might not even be taking biopsies. Avidity didn't take biopsies in their new arm. They did a biomarker arm.
Yeah.
But they already showed their drug worked.
Yeah, I'm much more impressed with KHDC1L.
Well, they want to use KHDC1L, but again, I can still imagine. Again, I'm an idiot, and I was told I was stupid in so many words. When I say I was told I was stupid, I'm just reading the look that I get. People are polite enough not to tell me I'm stupid, but they're such bad poker faces. It radiates that you're stupid out of, and I read that. I'm very attuned to that.
Yeah, you are. You're
I've been told I was
extremely,
stupid for
perceptive.
ages. Yes. I like it. I'm dumb enough to do a podcast.
Yeah, there is that.
Dumb enough to care about patients and what's good for them. So a biomarker only arm. Could they actually get to phase 3? Could they actually get through all of this without ever showing that their drug works?
Maybe. I don't think the FDA really cares.
I mean, at some point, how far can you get and then have it be pulled off the shelf and you realize that it's just that you're actually...
Well, it is a very expensive...
illusionists, magicians.
That's an interesting question.
Maybe Ursula Fisher will play and Woody Harrelson. They're still around. Jesse Eisenberg, we got the whole gang in, and we'll make a made-for-TV movie.
You need the SNL skit where there's one you missed, which is very appropriate to a triple immunosuppression regimen, and that's the steroid Olympics.
I was just thinking that the all-drug Olympics.
Yeah, the all-drug Olympics. Yeah, where the guy's arms fall off. You got to.
Ooh, he's gonna feel that in the morning.
Yeah, that's what you don't want to do on a clinical trial.
So we give this the uh-oh emoji.
Yeah, that's good.
All right. But seems like they will do everything they can to avoid showing methylation DUX4 target gene expression.
It sure does.
Data. But you know what? I may be wrong. And Amber's going to be talking at the World Muscle Society meeting in Hiroshima, Japan in October, and I'm sure she will have all that. I hope she has all the data.
Maybe
and
they'll
they show it
cobble
all
it together by then.
I hope they have all the data, and I hope it's spectacular. I truly do. But I gotta tell you, I guess we're just more cautious. We're very cautious. People say, we're very, very cautious. So I would love somebody to do a podcast on our data and rip it to shreds like this. Because one, I would address every one of their concerns. And two, I'd be amazed if they could rip it to shreds because we rip it to shreds before
We do a
ourselves.
pretty good job.
Good job. All right, so don't forget the way that this works. This is Epi321 mechanism of action is to methylate DNA and shut down DUX4. It's absolutely vital to show target engagement. But you know what's going to happen? You know what Facebook cares about? You know what everybody's talking about?
Did you guys hear about that car that runs on water? It's got a fiberglass air-cooled engine and it runs on water, man!
Right?
That's
Easily distracted. Easily distracted. Right? Breaking news. Epicrispr reports this is going to be here maybe we'll modify this and say,
Yeah, another one of my favorite.
Epicrispr reports first clinical evidence of increased lean muscle volume in patients with FSHD following treatment with prednisone.
I love that.
Now, I was thinking about this as I put this on here. The reason it's not valid cartoon, though,
Why?
because those freezers work.
Oh, that's true. And if they're not plugged in, they're gonna work.
He may have sold Inuits freezers and refrigerators
Everything
that
will
they
stay
didn't
frozen.
need, but at least they work. Anyway, I really appreciate you.
Anyone who's still listening, we appreciate you. You made it through all that.
We'll stop the share. Good times are had by all. You
yeah.
got to go catch a
In
bus.
6 minutes, I don't think I can
Out
do
of
it.
here. Thanks a lot.
Yeah.
All right.
You better not cut all my stuff.
Take us home.
Bigger.
Thank you for listening to the MyFSHD podcast, where we share with you the latest news and information on FSHD as we strive for a cure. Do you have a question for Peter? He'd love to hear from you. You can reach him at peterjones@med.unr.edu. Thank you for listening, and join us again next time for more news and information